Love is coming round for your pleasure. Push. There's no need. People go against the grain with the greed. I've been here before. It ain't gonna work no more. I've been here before. It ain't gonna work no more. Focus, your mind will be strong. No distractions. So called wisdom all year long. And then take action. I've been here before. It ain't gonna work no more. I've been here before. It ain't gonna work no more. I've been here before. It ain't gonna work no more. I've been here before. It ain't gonna work no more. Oh no 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 more. I've been here before. It ain't gonna work no more. Oh no more. I've been here before. It ain't gonna work no more. I've been here before. It ain't gonna work no more. I've been here before. It ain't gonna work no more. For your love. When I think about those nights in Montreal, I get the sweetest thoughts of you and me. Memories of love above the city lights. Ooh, I try so hard to take it, but oh Lord, my heart would make it. I just wanna stop and say what I feel about you, babe. I just wanna stop and never wanna leave without you, babe. I just gotta stop for your love. When I think about the way the world was served, I get the saddest thoughts of you and me. Memories of life and time goes on and on. Ooh, I try hard to forget it, but oh Lord, my heart would make it. I just wanna stop and say what I feel about you, babe. I just wanna stop and never wanna leave without you, babe. I just gotta stop for your love. When you close your eyes and you go to sleep.
Olá. Hello. Yes. Bom dia. Good morning. I have to read. Bom dia, good morning, buenos dias. Trilingue. No, I'm sorry, just 2 slides. That's all my repertoire in Spanish. Bem-vindas, bem-vindos, bem-vindas, welcome. Bienvenidas, bem-vindos. Okay, I'll stop the Spanish now. It is such a pleasure to be hosting the 3rd edition of this event, Placenta and the Development of Disease, with my colleague and friend. Sorry. I did something, Vinicius. Acho que yes, because the pointer. Yes. Yes, Tanya. So she's there. Yes. Thank you so much. Thank you all for coming. It's a pleasure to see a full house. And we are here at a different venue. We are in the university, but the campus went in a different venue. For me, it's the first time that I'm here, and it's a bit different, but it feels nice. Of course, it feels nice. It feels nice because of the air conditioner, but also because it's inside this auditorium. For me, it's very cozy, right? And I hope you feel the same. You feel comfortable, and so that we have a very productive morning and afternoon and discussion, right? First, I'd like to start with the acknowledgement. The main sponsor of this event is FAPERJ, and also CAPES and CNPq. CAPES in particular with the CAPES print scheme, right? That what made it possible for one of the speakers, for Jorge, to be here. And also we have our institution, the university, and sorry for the resolution, but Parque Tecnológico and this space in Novateca, it is free of this space. The university community can use, and it is free of charge. So it is a privilege for us, right, to take advantage of this. And I'm very happy and proud that we can kind of enjoy. And also our institution, our units, Institute of Nutrition and Instituto de Biofísica Carlos Chagas Filho, the postgraduate programs, and also the Conticom, which helped us with the graphical design, and a cooperation with Café del Rio, which was the coffee that you enjoyed outside, right? Okay. Also, I'd like to thank Carol and Alex for the support. Carol is there and Alex outside, the one that you register. And personnel from the Parque Tecnológico, Vinícius, Vanusa, and Kelly. Sorry, it was supposed to be and. Okay, food and drink not allowed inside the auditorium. During lunch break, we'll be serving sandwich and snacks, serving or offering, right? I think I'm not going to use this for now. Sandwich and snacks will be offered on the 3rd floor, which is the virtual window. Pode entrar. Pode, fica à vontade. And I think during lunch, of course, you're free to, to have lunch or to go outside, but I think it's going to be during lunch a good opportunity for networking. Not only a good opportunity because of the environment. It is very cold. It is very cool upstairs, but also cool because of the air conditioner, right? And after the event, around 3:30, we'll have refreshments and drinks for, a small, tiny happy hour. Okay, so if you're still here, I'm glad to join. Certificado, now em português, certificado de participação é fornecido somente para aquelas pessoas inscritas. Para as pessoas inscritas. Certificate of attendance will be available only for those who are registered. This is mainly for kind of the control for the university, because it was Tanya and I, we conceived as a discipline, right? So that people, of course, can enjoy the workshop, but also be, a commitment as well. Okay, now the workshop. We'll have 3 talks in the morning and 3 in the afternoon. And all talks we conceived as a 30-minute talk plus 10 minutes discussion. But this format we can rearrange, of course. Now we started a bit late, but since we're at home, so we are free to change whenever kind of the way we feel it and the way the discussions are flowing, right? This year's edition theme is lifelong health, maternal and environmental factors during pregnancy and lactation. I'm very happy and I'm confident that we will have very fruitful discussion, and we'll be dealing with environmental factors such as maternal diet and nutrients, maternal infection, drugs, and possibly other environmental factors that might affect microbiota, placental and fetal development, offspring metabolic health, and fertility. So I'm pretty sure that you all kind of enjoy, and hoping that we can possibly get, collaborations and other, ideas to different projects. So without further ado, yes, that's it. I'd like to introduce Jorge Lopez Telo as the first speaker. He is a junior PI at the University of Cambridge at the Physiology, Development and Neuroscience Department, Center for Trophoblast Research, right? So it is such a pleasure for you to be here, that kind of to have you here, of course. And again, CAP Sprint sponsored your visit to Rio. It is your first time second time in South America, but first time in Brazil and in Rio, right? The focus of his research is to understand the impact of genetically modified placental endocrine genes, right? And the impact on maternal metabolic health and offspring growth, right? And you're talking a little bit about this. And also, Jorge is not only, he's a very brilliant scientist, but his personal side, he really likes diving. Actually, he went to Arraial do Cabo last weekend, and so when he's here, he's safe. He said the water was a bit cold, but yes, but he was, of course, Arraial do Cabo, and also and you have pets, yeah. Yes, back in Spain. So he's based in Cambridge, but he's from Spain, and I think that's it. Thank you so much, and the floor is yours. Yeah, yes.
This one, right? Yeah. Thank you so much. Thank you, Tatiana, for the nice introduction, and thank you for the invitation. It's a great honor to be here with all of you. Thank you, everyone, as well, for coming to this seminar. I hope, I'm gonna show you 2 big projects that we have been doing at the University of Cambridge, and I really hope to convince you about what we are doing there. And I would like to start this talk always by trying to share my point of view about pregnancy. And from my point of view, pregnancy can be considered as an orchestra. So during the most important performance of our life, the placenta, from my point of view, let me just see. Okay. Yeah, it's a bit weird. The placenta is going to act as a chief conductor, and the different maternal organs, which are represented as the instruments playing in this orchestra, will receive no, sorry. Will receive different signals from the chief conductor, from the placenta, in a way that these signals are hormones, and these hormones will mediate changes in the different maternal organs. We know that, for example, placental hormones like progesterone, they can induce changes in the expansion of the pancreatic beta cell mass, and others like steroids like estrogen or progesterone can mediate changes in peripheral tissues of the mother, like the liver, the skeletal muscle, or the adipose tissue, to promote allocation of resources to the growing fetus. However, this vision of the orchestra is not as simple as I have just shown, and it's not only different organs and a chief conductor. In fact, from my point of view, this is like a more accurate vision of this orchestra, in which we are going to have what I call them visitors. And with visitors, what I mean is different microorganisms that will establish a symbiotic relationship with the mother in most of the cases. And I'm really interested in the role of the gut microbiota, specifically in the role of a specific type of bacteria. So who is the placenta, and who is the gut microbiota in this orchestra, and why these two conductors, from a point of view, are important in driving the pregnancy? So first of all, we have to introduce who is the placenta. So the placenta, from my point of view, is one of the most important organs that, well, we had in our life, and it's the most understudied organ. And it has multiple functions. It anchors the fetus to the uterine wall. It provides immunological protection. It acts as a detoxification organ. And most important, from my point of view, is that it is a super important endocrine organ, and it also transports nutrients and gases to the growing fetus. However, the problem occurs when the placenta does not work as it should, and this term is referred as placenta insufficiency. And we know that when the placenta does not work good, it can result in problems for the mother, like gestational diabetes or preeclampsia, but also problems in the development of the fetus, and also it can result in late onset diseases in the offspring, like obesity, diabetes, or hypertension, just because the placenta was not working as it should have worked during the period of the pregnancy. And despite we know all these situations, little is known about the role and identity of the different placenta endocrine factors mediating changes in maternal metabolism during pregnancy. Among the different genes that the placenta has, there is one gene that it really stands out during the period of the pregnancy. And this gene is insulin-like growth factor 2, or IGF-2, which is a paternally expressed imprinted gene. This doesn't work, but okay. This one. It is a paternally expressed imprinted gene, and with this means that this gene is actively expressed by the paternal side to promote growth of the offspring. Maybe. Okay. It's fine. Otherwise, I can point it out with my finger. It's fine. Let's just carry on. So IGF-2 is a paternally expressed imprinted gene, which will try to promote allocation of resources to the offspring. Which IGF-2 from the maternal side is normally silenced to avoid depletion of her own resources by this H19 imprinted gene. So IGF-2, after binding to the insulin receptor and IGF-1 receptor, promotes cellular growth and proliferation. We know that IGF-2 can be dysregulated in situations like gestational diabetes, preeclampsia, and in specific symptoms or like the silver Russell in humans. However, the vast majority of the knowledge that we have obtained about the role of IGF-2 is by the use of animal models. And we know that, for example, in the IGF-2 null animals, where IGF-2 is reduced, we know that fetoplacental growth is reduced. There is also alterations in the transfer of nutrients to the growing fetus, and there is a reductions in the formation of placenta endocrine cells. So with this background, we formulated our first scientific question. what is the role of placenta endocrine IGF-2 on maternal metabolic health? So for that, in the lab, we answered this question by getting the benefit of using animal models, and specifically we work with genetically modified animals. And for this project, we use gene deletion models, and we use the TBPA grid line. And as you can see here, hopefully, yeah, in this the green part, this one is where the crease is expressed. And this means that by using this type of animals, we are able to delete the expression of IGF-2 exclusively in the endocrine cells of the mouse placenta, leaving intact the expression of IGF-2 in the maternal decidua, as you can see here, in the labyrinth region, so in the transport region, and also in the fetus. So all the effects that you will gonna see now are exclusively mediated by a reduction in the expression of IGF-2 in the endocrine cells of the placenta. Due to time constraints, I will be only able to show the loss of function model that we have recently published, but also in the Q&A sessions or later during the coffee breaks, I'm happy to show or to chat about the gain of function model, which is not totally opposite models. So first thing what we performed, we worked on day 16 of gestation for this part of the data that I'm gonna show you, which is the period in the mouse when the mother is most glucose intolerant and insulin resistant during pregnancy. We had 3 groups to understand. We performed a group of non-pregnant animals to see how placental hormones mediate changes in the adaptations during the period of the pregnancy. And then we compared the results with the reverse parental cross. So we deleted the active copy of IGF-2 from the paternal side, as you can see here in this group. And then we compared our data with the reverse parental cross. And as you can see here, by doing this maintenance strategy, we are able to reduce the expression of IGF-2 in the junctional zone, but not in the labyrinth. I'm doing gene expression analysis in microdissected placental tissue. We are able to see a reduction of IGF-2 in the endocrine region. So using this model, what we have seen is that there are no alterations in the structure or in the size of the placenta in this model. There are no changes as well in the cellular population when we analyze this by histology. However, what we can see is that fetuses that were supported by these mutant placentas, they are growth restricted. Also, they are hypoglycemic compared to normal fetuses that were supported by normal placentas. And this is despite that there is no changes in the abundance of the glucose transporters in the labyrinth region. So there has to be a problem happening in here in the supply of glucose potentially. So in the lab, we have the tools to really understand how much glucose is transported from the mother to the fetus by performing placenta transport assays, by injecting a non-metabolizable analog of glucose. And what you can actually see is that dams that carry these mutant placentas, they fail to provide the sufficient glucose to the growing fetus. So this data suggests that when you have an endocrine problem in the placenta, the mother is unable to provide adequate amount of glucose to the growing fetus. So we then again, we move to really understand what's happening in the mother if this is the situation. So what we perform is the glucose and insulin intolerant test. And as you can see here in orange is the non-pregnant animals. And in purple, you can see a female that carry normal placentas with normal levels of IGF-2. And when you inject a bolus of glucose, you can see that these dams are glucose intolerant, and that's a physiological adaptation that happens during the period of the pregnancy to be able to provide glucose to the growing fetus. And also when you perform an insulin intolerant test, you inject a bolus of insulin, and you can see that these dams are insulin resistant. And to combat these adaptations during the period of the pregnancy, the pancreas needs to expand in size to provide more insulin. And these are physiological adaptations that occur during the period of the pregnancy. Okay, so what happens when we analyze or dams that carry the mutant placentas? These animals fail to achieve the glucose intolerant and insulin resistant state, and they behave as a non-pregnant animal, despite they were pregnant. And they also fail to expand the pancreatic beta cell mass. This is not only a problem of glucose. What we also observed is that, as you can see here, during pregnancy, the mother has to provide as well lipids to the in the circulation. They have to increase the circulating levels of lipids. And when we study our dams that carry the meat and placentas, again, the circulating levels of lipids were significantly reduced. So now that we can observe that these dams have a metabolic issue. So we wanted to understand who is the main driver of this problem. And of course, we manipulated IGF-2, and at the beginning we could think like, okay, is IGF-2 the main driver? So we checked the circulating levels of IGF-2 in the maternal circulation. And as you can see here, from a non-pregnant to a pregnant state, IGF-2 in the maternal circulation increased during as an adaptation in pregnancy. But what we could see is that IGF-2 in the circulating levels of the mother is not altered, suggesting that, okay, IGF-2 at the level of the placenta is generating an important effect, but it's not altered in the circulation of the mother. So we took a different approach. We performed an unbiased proteomic approach of the maternal plasma to really identify potential proteins that could be dysregulated in this model. And what we could observe is that a number of proteins that were altered, but especially there were like 2 that have been reported to be responsible of changes in metabolism. One of them, it was calcistatin, that has been reported to be altered in situations for diabetes. So potentially it could be inducing changes in the glucose and insulin handling of the mother. And the other one was prolactin, which, as I say at the beginning of the talk, is a hormone that is very important in driving changes in the pancreatic beta cell mass during the period of the pregnancy. And in fact, prolactin is at the level of a non-pregnant animal. So we then move again to the placenta to really understand what is happening in the communication of these trophoblasts or these endocrine cells of the placenta, and we perform a single-cell nuclear RNA-seq, and we really focus attention on the endocrine cells. And what we could detect as well is that there were a number of prolactins, but you can see them. There was a number. I know it's not working. Okay, yeah, well, you can see them there. There were a number of prolactins that were dysregulated between normal placentas and placentas with deletion of the paternal IGF-2. But aside of prolactin, there were also a number of genes that were dysregulated in response to the deletion of IGF-2, and many of them were involved in mitochondrial function and also in translation. Aside of that, we also performed secretome analysis to really understand what are the proteins that could be secreted into the maternal circulation. And we detected a number of proteins that were dysregulated, more than 200. And many of these proteins have been also reported to be involved in the control of lipids, like apolipoproteins or leptin receptors. So as a conclusion, what I can say from this part of the talk is that the placenta plays a critical role in signaling to the mother to adapt her metabolism to favor nutrient supply to the developing fetus. We can also conclude from this part of the talk is that defects in placenta endocrine function have negative consequences for maternal metabolic health. Okay, so we know already that the endocrine function and the IGF-2 is a very important gene or protein during the period of the pregnancy, but where we put now the gut microbiota in this orchestra. So from my point of view, the way that I like to explain this project is by comparing the gut microbiota with workers. And with that, I mean that we have trillions and trillions of bacteria in our gut. And these bacteria, they are going to work for us in the vast majority of the cases. So from complex nutrients, they are able to break them down into smaller molecules like short-chain fatty acid or organic compounds that will be used later by the placenta and by the fetus to support their growth. And among the different workers that we have in our gut, there is one that really stands out as the best employee from my point of view, which is bifidobacterium. And bifidobacterium, you can find them in different parts of our bodies. It lives in the gut, but also you can find it in the vagina, in the oral cavity, and it is used as a probiotic. And you can most of the times you can buy it in probiotics, like from pharmacies. And the cool thing of this work is, as I say, they are able to degrade complex sugars to produce energy in form of ATP, but also they are able to produce short-chain fatty acids like lactate and acetate. We also know that when this worker is bifidobacterium interacts during the period of the pregnancy with pregnancy hormones like progesterone. So bifidobacterium increase in late pregnancy, and the change in the abundance of bifidobacterium is aligned with the abundance of progesterone in the maternal circulation. And we also know that in situations like preeclampsia, the abundance of bifidobacterium in the maternal gut can be significantly reduced. So there is a connection between problems during pregnancy and the abundance of this bacterium. So with this background, again, we formulated the next scientific question, which was, does maternal gut microbiota control fetoplacental growth? So for that, we performed animal models experiments, and we took advantage of a quite a unique animal model, which is the germ-free animal. So if you are not familiar with this type of animals, they are quite unique because they don't have any type of bacteria in their guts. They live in isolators. They have never been exposed to any type of bacterium. So this type of animals really allows you to study the function the function of bacteria when you provide them to these animals, which is going to be a very direct effect. You give this bacteria, you get this result. So what we performed in these studies is we compared SPF animals. So these are normal animals that you can find them in any animal facility that they have like good microbiota. Then we compared the germ-free animals, which are these animals that I mentioned. And then the germ-free animals, when we provide bifidobacterium in 3 periods of the pregnancy, they 10, 12, and 14. And what you can see here in this graph is that when we provide orally this bifidobacterium, this is able to colonize the gut of the mother, suggesting that this treatment is good for colonizing the gut. And we provide it at a dose that it is commercially available for humans as well in terms of probiotics. And what we performed is, studies as well on day 16 and a half of gestation when placental growth is maximum. And also in a small cohort of animals, we performed the studies in late pregnancy on day 18 and a half to study the liver transcriptome, which is mature enough and resembles a kind of a newborn fetal liver. What you can see here is when you lack of maternal microbiota, in red in here, the fetuses are growth restricted, they are hypoglycemic, and the fetal liver is significantly reduced compared to a normal animal, suggesting that the microbiota is able to drive fetal growth. And more interesting, when we provide this bifidobacterium to the pregnant animal, bifidobacterium is able to rescue these 3 parameters. And now these fetuses that were growth restricted in germ-free conditions, when you provide bifidobacterium, they are at normal size. We then performed studies of the fetal transcriptome, and we detected more than 600 genes that were under the control of this maternal gut bacterium. And as you can see here in the pathway analysis, is that many of the genes that were dysregulated were involved in oxygen transport, but also in the control of steroids, lipids, and carbohydrate as well. So this really, we found that there was an effect happening in the fetal liver, but what is happening at the level of the brain? And this is the new study that we are trying to close now. And we know that, the oxygen is very important in driving fetal growth. And in fact, there are situations like, for example, HIF2 alpha that, it is also expressed in normoxic conditions, and it is an important transcription factor that can drive fetal brain development. And what we could observe is that at the level of the protein, when you provide bifidobacterium to the mother, we managed to get a stabilization of the HIF2 alpha in these germ-free animals. But also what we have observed is that signaling pathways like AKT or STAT5 were altered when you provide bifidobacterium to the pregnant animal, suggesting that bifidobacterium is able to remote control the fetal brain development. What we have also observed is that glucose transporters, the GLUT1 and GLUT3, and also the solute carrier 37A4, which is involved as well in the transport of glucose to the reticulum, was significantly altered when you provide bifidobacterium to the mother. So now we wanted to understand, okay, what's happening with the placenta, because we give a treatment to a pregnant animal, and what is happening in the interface between the mother and the fetus. And what we could observe is that when you lack of maternal microbiota, placental weight is significantly reduced, and this is mainly due to a problem or a reduction in the abundance of maternal blood spaces, and this parameter is not restored when you provide bifidobacterium. However, what we could observe is that when you provide bifidobacterium, there is a thinner or a reduction in the barrier thickness of the placenta, which this means that you want to have a thinner barrier thickness, so this will help or facilitate the exchange of nutrients and gases between mother and fetus. And we know that one of the drivers that can modify the barrier thickness of the placenta is IGF-2. So what we perform in this qPCR analysis of important genes that have been reported in the control of the barrier thickness, but also in the development of the transport region of the placenta in the labyrinth zone. And in fact, what we could observe is that IGF-2, so the gene that I previously described, is significantly upregulated when we provide bifidobacterium, and DLK1, which is another paternally expressed imprinted gene that promotes fetal angiogenesis and allocation of resources, was significantly elevated when we provide this bifidobacterium. And again, what we observed is that the abundance of the glucose transporter when you provide bifidobacterium significantly increased. So now what we are again trying to finish another story is what's happening with the endocrine function. We have seen that bifidobacterium from the maternal gut is able to remote control fetal development, and it's also able to control the transport region of the placenta. But what is happening with the endocrine function? So as you can see here, when you provide bifidobacterium, there is an increase in the abundance of certain types of prolactins. But we have gone even further. So we have generated a map of proteins that are under the control of bifidobacterium. So there is more than 400 proteins that are under the control of bifidobacterium. So when you give this bacterium to the mother, bifidobacterium can control these proteins, and many of them are involved, as you can see here, in metabolic processes, translation, mitochondria function, or ribosome function as well. So as a conclusion, I hope I have convinced you that the gut microbiota, it is an important player controlling pregnancy and fetal outcomes. It can control placental weight, and when you and of course fetal growth. And when you provide this bifidobacterium, the endocrine function, but also the transport function of the placenta is controlled by this bacterium, and this has important effects in fetal growth and fetal glycemia, and in the formation of fetal organs. So we can conclude that the maternal gut microbiota during pregnancy, including bifidobacterium brief, modulates placental morphogenesis, placenta endocrine function, and fetal growth. And this is the conclusions. I would like to say that, I mean, I'm editing a special volume in this journal called Veterinary Science. If you are interested in submitting any project, or you have any potential project that you would like to submit it, I can say in here that it is for free. You don't have to pay. I know that MDPI, they always charge you for a lot, but we have a number of papers that are for free. So I know, I mean, I did my PhD in Spain, so we know how hard it is to get funding. So we can chat about that. If you have a potential paper and you are interested in submit it, it can be brief communications, research articles, or reviews. And with that, I would like to thank all the people involved in this project. Amanda Sferrucci Perry Labs, of course, because it was a very long project, the first one, more than 8 years. And the whole lab, Lindsay Hall, for the microbiota, collaborators, and also the funding. I forgot to put in here, but as well, and this is something I would like to share. Well, chat with all of you, especially with people that are in here, because they are, I mean, I think like, yeah, there's young people in here, and I did my PhD in Spain, and then after that I relocated to UK in Cambridge. So there are certain grants that as a foreign you can apply to do your postdoc in abroad in the UK. So if you are interested on that, we can chat about that during the coffee breaks. Because I think, yeah, if you want to move abroad, this fellowship that I had from the Royal Society that supported the first project is a quite unique opportunity to be able to go abroad. So yeah, you can just approach me in the coffee session, and thank you for the fundings, and thank you again, Tatiana and Tania, and all the love from Tatiana that have been amazing this year. Those just no these days. Maybe yes. And thank you very much for your attention. I don't know, this one? Go.
So a very nice talk. Thank you. A lot of information. So I was thinking,
I know. Sorry.
you must be tired. So I'm trying to combine the talks, and I was thinking a lot about hematopoiesis and erythropoiesis, because the second part you focus a little bit more on the liver. And liver at these time points is crucial for produce the babies erythrocytes. Yes. So I was thinking, have you take a look at that? And this could also be related with the fetus growth as well, because he needs his own erythrocytes. And also the brain. I was thinking about the hematopoiesis, the myelopoiesis systems. The microglia also control. There are some papers that is important for the growth of the brain as well. And they also should be came from fetus liver.
Yeah, so I mean, this is funny because so in our lab, in Amanda's lab, there is one postdoc that is very interested about iron homeostasis. We have just in fact put it yesterday a preprint about high fat diets and iron homeostasis and how that this relates to fetal growth. It is interesting because in the so when we got the RNA-seq, if you check the list of genes, there are many genes that are involved in iron homeostasis. So, and the idea is that now when we got too many pages to get it out and published, but the idea is that for our next project we are going to look more carefully into the transporters of iron and what is happening in the metabolism of iron, because if in the liver RNA-seq of the fetus there are changes related to iron homeostasis, there should be a change happening in the transporters potentially. In the transporters of iron in the placenta. And I mean, so yeah, iron is in part, there is in the metabolism of iron, there is a connection with the gut and liver. So I really believe that there is potential iron changes in this model of the gut microbiota. But yeah, I hope maybe in the next 2 years or in next year I come back in here and then I give you an answer of that. But yeah, it is a project that we are gonna do now by assessing the different transporters in placenta and also in fetal organs.
Thank you.
Thank you.
Just to, this is about the IGF-2 model, right? That you're mentioning.
The
Yes.
iron?
Yes.
No,
it's
no.
that.
This is more in the microbiota.
Okay. Okay.
So in the IGF-2 we have not looked at the iron.
No, because what I was going to ask is about because you showed lots of genes transcripts related to mitochondrial function, right? And maybe suppressed, decreased, I'm not sure. Loss of function of mitochondria, because an enzyme crucial for heme synthesis is a mitochondrial enzyme.
Yeah.
So maybe there is a connection between the synthesis of heme and mitochondrial functioning in both models actually.
Yeah, I mean,
so
In the underexpression in the IGF2 model, definitely there are problems in mitochondria. We also check the PPR gamma, and it's altered in this model. We have done more mitochondrial studies, but in the overexpression model, when you are in the opposite model, but more looking into the placenta maternal liver communication, and this really highlights that there are problems in the mitochondria of the liver. With problems in the oxphos, and I think it was in the complex 1 leak, and also in NAD metabolism. So there is, of course, a problem between IGF2 and mitochondrial function.
Thanks, Tatiana. Jorge, that was fantastic. I think you know I'm going to ask you about the brain.
I know, and that's why I put it as well. I mean, so maybe you can give me feedback, because I'm not a brain person.
It's fascinating to see the effects that you've seen. I'm just wondering if you've looked at in any more detail at specific areas of the brain that may be impacted, and using single cell, you could do some really cool things within the brains of those fetuses or newborn mice.
Yeah, this is the annoying part of this project. So we are not a brain lab, and we collect brains like, oh yeah, let's see what happens just in case. But unfortunately, we don't have, like, for example, to do a structure, which would have been nice. And I think that's the problem that we are going to face probably when we send that for publication, because we cannot say, okay, the problem is happening in this, or the potential proliferation is happening in this specific region of the brain. We had to stop that project in that with this data. We have as well more about metabolites. There are specific changes in different, especially amino acids. But unfortunately, we had to stop there, and there is no way that we can repeat experiments, because we did it in a different lab, and we don't have more access to the germ-free facility. So annoyingly, very annoyingly, I cannot tell you more about that.
So, I mean, the other thing is that, with the germ-free mouse, it's a fantastic model, and you're using it really well. The question I have, though, is that, taking one bacteria and putting it in is very different from what happens, because all of them are interacting with each other, right? So I was just interested to get your insight as to, what those interactions might be, and what you might be missing by just going back with a single inoculum.
Absolutely. I mean, that's like, as well, when we published this first paper, it was we had to write a massive section of limitations of the study. I really like the germ-free animals as a proof of concept, or as a way that you really understand the function of a bacterium. But at the same time, I have to disclose, and I have to say that it is not an organism that is similar to a human. Of course not. And we have so many different types of bacteria, and potentially within the bacterium, when later interacts with other microorganisms, maybe you don't see that effect. But at the same time, what we have to really to understand microbiology and the gut microbe, you need to do this type of studies as well. Okay, what's happening? Isolated this bacteria, this is the function, and then you start combining with many others. So I think that there needs to be room for the germ-free studies. There is always a limitation as well. It's like when people study with antibiotics. Okay, you provide antibiotics, but at the same time, you are changing hepatic metabolism. And people don't, you have to be careful with this type of studies. So I like it, because you can really see how many effects you can do, but at the same time, you cannot be sure. What we miss in this study, I mean, you change or you improve your science by doing science. What we miss is the SPF animal, the normal animal, with a supplement of bifidobacterium, which potentially that's what we should have done. But then it's too late. Now you have done it, and now you cannot repeat it in the same conditions. But in the future, if someone is interested in doing that, I really recommend you to do that, so you will not have headaches with reviewers, as we had. I hope you were not one of them. Okay, thank you.
Jorge, congratulations on the talk. It was great, your work. I have one question. It's more of a curiosity, because kisspeptin is highly disregarded as to its effects. Potentially, kisspeptin derived from the placenta. I don't know in the mouse placenta, but in the humans, they have a huge rise in kisspeptin levels throughout the end of pregnancy. And kisspeptin do modulate maternal metabolism in general, and you have receptors for kisspeptin on the junctional zone. So have you considered to investigate whether you may have like dysfunction of kisspeptin on your junctional zone model that you just showed?
We have not checked kisspeptin. I know Amanda has mentioned that as well a few times, but we just didn't have time. I think the good thing of, I mean, we did a single cell nuclear RNA seq, which that gives you a lot of information. And I think, there's something that we can check at the cellular level if there are changes in the kisspeptin or kisspeptin receptors. I cannot give you an answer because we have not done it. But I may go later in the Excel and check that. Because there is connections and kisspeptin drives a lot of changes in metabolism.
Maybe be something there that maybe kisspeptin.
Absolutely. Yeah.
Okay, thank you.
Thank you.
Okay, nice. No? I have one. So what is, have you looked at for the microbiota results model? Have you looked at the metabolites?
Yes.
Because the reason I'm asking is because we have discussed a bit in a paper recently published, right? That the authors used antibiotics, and they kind of maybe showed that specific short chain fatty acid would be the drivers.
Yes. So we in the first study we performed metabolomics of the materno-fetal, and what we observed is that acetate was altered when you provide the feed bacterium, was increased. Then we also performed metabolomics of the placenta and the liver, and there were like changes. It's more between the SPF animals and the germ-free, but it metabolized like carnitine. And now in these new studies that we are trying to close, we have seen that in the junctional zone, there are two metabolites that are altered, and they are increased when you provide the feed bacterium, which is lactate and taurine. And then in the fetal brain, there are 9 metabolites, amino acids. Some of them, I mean, I will have to double check. I don't have it on top of my head, but alpha-ketoglutarate was one. Valine. There are a few of them, but especially related to amino acids, and also the transporters of amino acids are altered. So there is definitely, I think it's a combination of multiple factors driving the changes that we see in the fetus.
And what about maternal circulation?
We have not.
You haven't. Okay.
Annoyingly, we didn't. We should have measured it, but no, we couldn't.
Yeah, because in my point of view, the short chain fatty acid, of course, some amino acids, tryptophan, for instance, but short chain fatty acid might link to brain develop, of course. Yes. So maybe even if it's only one bacteria, but if it kind of resembles the profile of the metabolite profile, so I think
absolutely. I think it's the combination of that, like short chain fatty acids, metabolites. Potentially, I mean, yesterday there was published a paper about bacteria extracellular vesicles going to the fetal side. So I think it's you have you induce small changes in the gut, in the intestines, and that later communicates with the different organs of the mother, and then again you have an explosion of endocrines and metabolite signals that change everything.
Amazing.
So.
Okay, thank you.
Thank you.
Thank you so
very
much. Yes. Yes. Moving on to the second talk of this morning. Vinícius, yes. Você pode ver o pointer também? Por favor. So I'm going to introduce this second speaker, Marcos Gama Almeida. He's a PhD student from my lab, and so it was not very hard to convince him to come here to speak. And so Marcos is in his third year of PhD program. at the postgraduate program in nutrition at our unit, and his interest is to investigate the effects of COVID infection during pregnancy, but not only during pregnancy, but in the severe case of the disease. And so that's what he's going to talk a little bit about this today. Also, Marcos, not only for, kind of I was the one that asked for more personal stuff. He really likes to read, and he's always looking for a new book. And he has a pile of books kind of waiting for and one of it was for me we had this gathering for the lab this once, and the amigo culto, I don't know how to say this, amigo culto in English. Hi? No, yes, so the one that we give gifts, we kind of pick a name and then we give gifts. But anyway, it was to exchange, to give books. Yes. So to give provide books as a gift, and he gave me a Sex, Mitochondria, and Suicide from Nick Lane, and so he's a very kind of important researcher from London, and so this not only novels but also scientific and stuff. So thank you so much, Marcos. Yeah.
Just checking.
Eu acho que pode usar esse novo. Não, o novo.
This one is...
Tá melhor. Tá melhor, então. Beleza.
Okay, so good morning, everyone. As Tatiana said, I am one of the PhD students here from the university. Right now, I'm going to present what is part of my PhD project. The title is COVID in Pregnancy. I'm going to show some biomarkers of placental dysfunction in neonatal outcomes. So I cannot start this presentation talking about COVID without remembering us that in recent decades, Brazil has frequently dealt with arbovirus epidemics or some of these diseases that the mosquitoes is the main carrier of the virus. So dengue, Zika, and chikungunya impose a significant burden on the population and the public healthcare system. If I may remember, in the last edition of this workshop, we saw a presentation about the relationship of Zika and the brain development in the fetus. So more recently, in 2019, SARS-CoV-2 emerged following an outbreak of pneumonia in China. And with a higher potential for transmission and pathogenicity, it evolved into a pandemic. So here to us Brazilians, we also were looking for if this new disease has the same impact as the ones that we already have here. So despite the fact that the majority of affected individuals experience some kind of asymptomatic infection, a significant proportion has the potential to progress to severe illness, thereby increasing the demand in public health system. That is kind of the main problem that we saw in the outbreak of the pandemics. So right now, globally, COVID has surpassed 700 million cases and near 7 million deaths, so it's about 10% the fatality. And Brazil has the second highest number of fatalities with more than 700,000. And despite of we are seeing some kind of the lowest levels of death rates in new cases since the outbreak, we still have to deal with the people and the impact of this new disease that is not ended in our health public system. So to introduce the coronavirus disease, it's well known that the replication of the coronavirus affects several cellular factors, which induce some structural and functional changes in cells that is leading to tissue damage. And its ability to infect these different type of cells relies on the expression of the angiotensin converting enzyme 2, or ACA2, which is the receptor necessary for binding and entry into target cells. So when we have the infection of the virus utilizes this receptor to enter into the cell. I'm going to point it. And by the onset of the infection, as the virus utilizes this receptor, we already have some dysregulation of the renin-angiotensin-aldosterone system, which may cause tissue injury, inflammation, vasoconstriction, and once inside the cell, we also have some dysregulated immune response that we believe that is the major cause of this extrapulmonary damage. So when the virus is inside the cells, it can trigger some physiological reaction that we call, during the pandemics, we call this a cytokine storm, or some kind of hyperinflammation that causes an inflammatory cascade, and that will cause this tissue damage, and specifically to the organ that this infection occurs, it may cause some multi-organ dysfunction syndrome that it goes some kind of extrapulmonary effects, because in the beginning of the pandemics, we thought that COVID was only some kind of flu symptoms. And this is the major problem that we have when considering the long-term health for the people that got the severe spectrum of the disease. And if I said about organs and considering the main subject of this workshop, I may ask, what about the placenta? If COVID has the potential to infect and to cause some damage into different kind of tissue, what about the pregnancy state in the placenta? So the most recent papers show to us that when we have placental infection by SARS-CoV, we may encounter some increase in stillbirth or increasing preeclampsia, as we see here. And the direct effects of the virus in the placenta may cause infiltration of maternal macrophages and trophoblast necrosis, while the decidual side of in the decidual side you see the activation of natural killer and T cells, and some kind of gene expression associated with preeclampsia. And this is when we see the virus infecting the placenta tissue. But as I said earlier, COVID also triggers some kind of physiological response known as the cytokine storm or some hyperinflammation. And when, even in the absence of the placental infection, SARS-CoV-2 also induces some inflammatory changes observed in the decidua and the placenta, and these changes may be linked to increased risk of preeclampsia. Of course, all this data is very new because we are right now observing these effects, and these new studies are coming to us into this year. So just to remember, during pregnancy, considering pregnancy and the onset of the COVID infection, the SARS-CoV-2 infection, during the pregnancy, we have several changes that have the potential to predispose pregnant women to complications resulting from any kind of infection, not only COVID. But just to cite some of them, in the cardiovascular system, we have a natural increase in cardiac output and elevation in maternal heart rate to ensure the fetal nutrition. We also have a state of hypercoagulability because we know that the mother increased the plasma volume, and we also recognize pregnancy induces a state that is similar to diabetes because we see hyperinsulinemia and hyperglycemia as a way to ensure greater availability for the placenta and the transfer to the fetus. So this is specifically roles, these physiological roles that occurs during pregnancy may represent some risk if the pregnant women got the severe spectrum of the disease from COVID. And COVID-19 infection during pregnancy increases the risk for maternal and fetal complications. And considering that COVID-19 is not in that pandemic state, but evaluating to the endemic state, cases continue to occur worldwide, And research is needed to improve this clinical management at this specific group. Here in Rio de Janeiro, we already had a guideline to guide these pregnant women about the risk and how to care if they got COVID, or if they got the coronavirus disease. But of course, this isn't enough. And what I can say about this specific population that is part of my study is that the data provided by the Brazilian Obstetric Observatory indicates that until May 2022, we had at least 24,000 cases of SARS-CoV in pregnant women in Brazil. And the lethality of COVID-19 among pregnant women in Brazil was approximately 10%. This is significantly higher than the global population's average, which stands about 2.8 or near 3%. It's about 3 times bigger. So in Rio de Janeiro, we had the highest fatality rate in Brazil of approximately 11%. So this is why I think that specifically to us, that we are dealing with the local population. This is an important topic to study. And this brings me to metabolomics. That is the tool that I use in my project, and how metabolomics is really useful to this kind of infection and specifically disease, because as I said earlier, we had a multisystemic nature of COVID-19, and this demands some kind of integrated approach to comprehend the molecular mechanisms associated with this COVID-19 pathogenesis. And to those who are not familiarized with the metabolomics, the power that metabolomics had lies into this ability to recognize endogenous and exogenous metabolites within any kind of biological samples. Particularly to us, we already use plasma samples and saliva, but you can use pieces of tissue, blood, or anything, any kind of body fluids. And the prepare of these biological samples permits to us measure the concentration and identify perturbations of biochemical pathways associated with qualitative and quantitative metabolic changes. So when we utilize these body fluids or any kind of biological samples, may allow to us, utilizing proteomics, metabolomics, or lipidomics, to find some definition of molecular pathways and pathogens, or maybe, I don't know, local damaged tissue or any specific metabolites that represent some organ. Biomarker for prognosis. We may create new info about drug engineering or drug discovery, or maybe even creating info about vaccine development. So this brings us to my study object, that is to investigate the metabolic alterations induced by severe and non-severe COVID-19 during pregnancy, possibly associated with pregnancy outcomes and placental responses. I just need to remember that these are ongoing projects, so I don't have all the results that could tell us a good story, but it's something that I think that is already good enough to be more to present to today. So in this study, I had 3 main groups that we called them pregnant women. Of course, all the 3 groups are composed by pregnant women. The first one, we have a symptomatic pregnant women, but they got negative SARS-CoV-2. They are like my control, but they are symptomatic to flu symptoms. My second group are, of course, pregnant women, also symptomatic, but they got the positive results from the SARS-CoV-2 PCR. And the last group are also symptomatic and positive to SARS-CoV, but they are on the severe side of the spectrum of the disease. This third group right here are the ones that needed the ICU hospitalization. They needed to be into intensive care. And we did the collection of clinical data in plasma upon the examination or the admission to ICU. And we processed this biological sample, and got to the acquisition of the spectrum, the nuclear magnetic resonance. So after we got the spectra, we did the data processing and metabolomic analysis to see if any metabolites tell us if anything is different is happening among these groups. It is important to say that as I have two groups that people are not into some kind of hospital facility, and one of them is into the ICU or intensive care, these first two groups that are the negative COVID and the positive COVID but non-severe, they were recruited at the Diagnostic Triad Center associated with the Nucleus for Confrontation and Studies of Emerging and Re-emerging Infectious Diseases here at the university from March 2020 to May 2022. So it's between a period that we already started the vaccination, but great part of these pregnant women did not take any kind of dose from the vaccines. In the group of the severe pregnant patients, we have 13 pregnant women that were admitted to the ICU at the São Sebastião State Institute of Infectious Disease, also here in Rio de Janeiro. So at first, we look into the demographic and clinical characteristics. We see that age, gestational period, and presence of comorbidities did not show any statistically significant difference, but regarding this gestational period that is around here, it's worth noting that in the third column, the positive severe group, the ones with that severe disease, only 80% of them were in the first trimester or the early stages of pregnancy. And this may suggest an increased risk of severe outcomes when the disease occurs in the later stage, because if you see here, when compared to the other two groups, there are almost at least 30%, and this only 80% of them on the first trimester. And the frequency of symptoms did not exhibit a clear difference among of groups. Of course, I did not analyze the symptoms of the severe ones because they are already into intensive care. And first, when we had the acquisition of the spectra and look at them at the principal component analysis, showing that this graphic shows each point here is one of these women's. The negative symptomatic are the red circles. The positive symptomatic, but in the non-severe group, are the green ones. And the blue circles around here are my pregnant women from the severe group. We can see in this graph that we have a clear separation from the group that is considered severe from the other two. It allows me also to conclude that in the case of mild symptoms or non-severe COVID, it's kind of seems similar to the flu symptoms or negative to COVID when I talk about the metabolic profile. And when we consider only the COVID positive women, the non-severe and the severe are the red circles and the severe are the green ones. We also see a clear separation between them. And this shows to us that these severe women had a different profile, metabolic profile. And when we look to the specific metabolites that we are able to identify in the spectra, they tell us a story that I called some kind of insulin sensitivity issue and energy metabolism. I will show right now, if you look to these graphics, we have here glucose and lactate. This first column is the negative symptomatics, in the middle the positive, and the last one, the black dots, are the positive severe. We are seeing that in my third group, in the severe pregnant women, we have higher levels of glucose and higher levels of lactate. Of course, higher levels of glucose is not a desirable outcome, but elevated lactate is commonly found in diabetic patients, and this could indicate insufficient oxidative capacity and also hepatocellular damage. And it may allow me to ask if these higher lactate levels in severe COVID-19 during pregnancy may also indicate altered placental energetics. And when you look for some of the amino acids that we are able to identify, the BCAAs, the branched chain amino acids, The previous study shows that BCAAs were already increased in women who developed the gestational diabetes, being the most common and most important abnormality of amino acids in metabolism in this gestational diabetes. So as we see here, valine and isoleucine, who are two of the three branched chain amino acids, it looks to be increased in this severe group. And as I said, BCAAs is the most common and most important abnormality when you talk about the gestational diabetes. Other metabolites who also maybe talks about the same issue, that is insulin sensitivity or energy metabolism, was the metabolites related to lipid metabolism. Here we are seeing the signal from triacylglycerol, LDL, and VLDL. And as we see here, the three lipoproteins are usually high in my positive severe group. As you see by the black dots, they are always high when we compare to the other two groups. And this increase in these maternal lipids across the gestation is also a common result of insulin resistance and a predictor of gestational diabetes. As I may remember, we don't have any of these pregnant women from the severe group diagnosed with gestational diabetes or any kind of diabetes previous to the pregnancy. We have other metabolites that also show some curious information. Acetoacetate, that is a ketone body, used as energy source when glucose is not readily available, is commonly found increased in diabetic patients and usually indicates poor insulin sensitivity. And again, we also saw that acetoacetate is increased in my severe group. And acetate seems to be lower. Acetate is one of the major short chain fatty acids alongside with butyrate. And the depletion of acetate, these decreased levels, as we see here. Acetate, as I said, is the main intestinal microbiota fermentation product. It's related to some beneficial health effects, but is also associated with diabetes metabolism, while this may reflect modification in the gut microbiota in these women. Also, some recent studies indicate that acetate, alongside with butyrate, improves beta cells metabolism and mitochondrial respiration under oxidative stress. And I may remember that COVID also impacts in the renin, angiotensin, aldosterone system and the circulation of the blood pressure and things that are related to this oxidative stress. And we also were able to identify some amino acids that maybe to me allow me to hypothesize about some kind of tissue damage that I talk about in the introduction. Here I'm showing lysine and here tryptophan. And lysine is clearly decreased in my positive severe group. And when I looked for lysine levels in other metabolomic studies, I saw in the literature that reduction of lysine is observed in the early onset of kidney damage and hypertension. That it also allows me to see that maybe these severe women are experiencing some kind of tissue damage in the kidneys. And the tryptophan, that is slightly increased, but got a statistically significant difference. When I searched for the tryptophan levels and the impact on gestation, I saw that this impaired placental perfusion are associated with inability to regulate the tryptophan-kynurenine pathway during pregnancy, and these individual differences in maternal tryptophan and or kynurenine during pregnancy could have substantial long-term effects on fetal brain development. So to me, this is an important metabolite to see if these may interfere in the fetal or neonatal outcomes. And here I also saw some kind of dysregulated one-carbon metabolism and inflammation, here representing glycine and histidine. They are altered in my positive severe group. And I should mention that SARS-CoV-2 is known to hijacks methyl donors and one-carbon metabolism for viral replication. So this may be a virus response to guarantee the replication. And decreased levels of circulating histidine, as we see here, the decrease in my positive severe group, is also linked to elevated blood glucose because histidine is possibly involved in hepatic gluconeogenesis. And I also saw one less metabolite that I call N-acetyl of glycoprotein that seems to be higher in my positive severe group around here. And an N-acetyl of glycoprotein is a marker of inflammation and is also commonly and significantly elevated in women with gestational diabetes, while also being associated with cardiovascular disease. So this is an important marker to not only cardiovascular, maybe tissue damage or inflammation in general. So this is my previous results, and overall, if you are having the same conclusion that I had, these early results may suggest that severe COVID-19 appears to induce a metabolic state similar to what we see in gestational diabetes, even if these pregnant women were not previously diagnosed with diabetes. So diabetes in pregnancy strongly predict perinatal complications, and considering results, it is possible to hypothesize that severe COVID-19 during pregnancy may impact in a similar way. So future analysis such as the high pressure liquid chromatography metabolomics or lipidomics, the acquisition of more data from these gestational and neonatal outcomes may help elucidate the impact of COVID-19 during pregnancy. As I may remember, we collected the data from these women between 2020 and 2022. So right now we are able to ask them again to answer some follow-up questionnaire to see if they had any income in the outcome, any gestational or neonatal outcome. So this is some of the future results that I should probably, I don't know, present in the next year edition. And I may not end this presentation without recognizing the ones who supported me in this project, especially Tatiana, my advisor, and Terezinha, who are the one that allow me to use these plasma samples, and other collaborators such as Alexandre, Iranai, Luisa from the Microbiology Institute, and the people from the National Center of Magnetic Resonance, and also CAPES and FAPERJ for funding the financial aspects of my project. Thank you for your attention.
Thank you, Marcos. Questions?
Thank you. That was a really, really interesting presentation. The one question I have, seeing all of the metabolic differences that you see is really fascinating. I'm just wondering how much of it is due to COVID, the COVID virus, versus how much of it is due to the severity of the disease and the symptomology. So, you know, could you look at another disease which has similar symptomology, severity, and see whether you see the same metabolic effects, to see whether it's specific to COVID-19 or whether it's just something to do with the severity of the disease, a severe respiratory disease, for example?
We know that in the severe spectrum of disease, we already published a paper that were not in these pregnant women, but in general people from the hospital, and we had two groups. One of them were fatal patients and survivors, but they all are the severe spectrum of the disease. And we know that the one carbon metabolism, specifically choline-related metabolites, seems to be very specific to COVID, and also some amino acids, just like tryptophan. Not like I showed here, tryptophan in COVID, in the severe state of the disease, seems to be very, very, very decreased in the fatal patients, and it seems as a marker of the progression of the disease. Also this choline and choline-related metabolites, because it seems it's really important to the viral replication. So I may choose these ones as some specific COVID response, I also have my doubts about the effect of the internalization, the ICU hospitalization, because it's already changes the diet, and I don't know, some medicine that they are taking right there. And this also impacts the result that I had, but we don't have any way to control, to control this. So I think that one carbon metabolism in amino acids may be the main aspects of severe COVID when not pregnant women, but general severe COVID.
Great. Thanks very much.
Thanks, Steve. We have data on viral load, right? So that's the next steps. So it's going to kind of try to associate the magnitude of the effect to viral load, as you know, taking from the PCR, and also the duration of the symptoms. So then we can see acute respiratory infection, not necessarily COVID, but taking kind of picking these metabolites that we already saw in a different group might give a hint that this might be some specific to COVID, severe COVID infection. Yeah, right.
Thank you, Marcos, and congratulations. You got amazing data. I was just thinking while you were presenting, do you have the opportunity, for example, because the way that you have described your groups, from my point of view, what I was missing is what's happened with patient that it is positive in COVID, but asymptomatic as a real control, normal baseline level. If you have the opportunity to analyze that extra group, and that will give you, okay, different grades of COVID. That was one point. Then the other points you have mentioned about like tissue damage, but can you, with your biochemistry or with analysis, can you be more specific about what is the tissue that is damaged? Is by analyzing, for example, hepatokines, ALT or AST, or is it like more muscle damage, like creatine kinase, by measuring that? And in the last one with the diabetes, are you, because you showed that, yeah, you have hyperglycemia, but is that related to hyperinsulinemia or changes in fructosamine, or? Yeah.
Well, the first question, these women, all of them presented like voluntarily to do the exam at the center of diagnostic here. So they all already had the doubt if they have the COVID. So we still had some, I don't know, about 10 women that didn't show any kind of symptoms, but we excluded them from the samples to not create any kind of confusion. And because they all presented to do the exam, they all thought they had COVID, so most of them present any kind of symptoms. We don't have any subject who had COVID and got any kind of symptoms because they just don't show up to the hospital to do any kind of checkup in the virus. And right now we are trying to get access to the blood tests that these women may have during this follow-up of the disease, but we only got the ICU hospitalization into this severe group, and we don't know if the rest of them had any kind of information to give to us. We are trying also to get the information if this pregnancy everything is fine, if the fetus or the newborn is fine also, because it may relate to the ongoing of the disease. And to the specific tissue damage, we are counting on these blood test parameters, such as AST, ALT, that we have to into the severe group. But when we are dealing with human subjects, it's really hard to do these steps, because if you not ask them in the first moment, you have to do another, I don't know how to say, you need to ask to the ethical committee to ask them again, ask them if they want to participate and give us more information. So we are in these kind of steps right now. But I think if you had this, you probably should confirm the physiological damage, because other papers already published that it's clear that some specific organs like liver and kidney it's the most damage in the severe spectrum of COVID.
Yeah. Thank you.
Congratulations on the presentation. Very nice. Wonderful data. I was just wondering if you took into account gestational age when you're performing your analysis, and how are you going to control for gestational age in your data?
We took that in consideration when we are doing the analysis, but my sample in the severe group is really too small. I can show you, we only have 1 in the first trimester, 5 in the second, and more, I don't know, 7 in the third trimester. So if separate them, you just got a too small sample size that incapacitate us to do any kind of statistical comparison among the groups. But this is really an issue when you talk about the metabolic state of these women, because we know that the first and the third trimester seems to be really different from each other, the fluctuation of these metabolites. But this is something that we take in consideration. But at this state of the research, we are really don't know how to deal or even how to discuss this. It's going to be some kind of minor issue for my research. Also alongside of the ICU internalization, or maybe if these women got into the mechanical ventilation, this also changes the metabolic profile. This is also an issue that we are going to deal when we are going to publish these papers.
Okay, so other 2 questions very fast. So did you think, you and your advisor think of measuring cytokines in the blood, the cytokine profile, as well as some hormones like a human blood HPL, or even estrogen and progesterone?
Well, we wish for to dose these hormones, but we already received the samples from the blood samples when the moment that these women arrived at the hospital. So right now we don't have how to collect the blood samples and relate it to this period.
So you have no more samples?
No, we already use the plasma to we have a little more, I think we have some kind of 250 microliters. I don't know if this is enough to do any hormone dosage, but we are looking into the hospital to see what they have and what they can give to us. I think the as Jorge said, the ST, LT, some hepatometabolites we have, but hormone, I doubt they have, because hospitals usually don't make this kind of blood tests.
Yeah, just more like question you could say. And how did you collect placenta from those?
No.
No, actually, he wanted to do everything, not everything, but we are kind of, this is a multi collaboration project. So we have people dealing with cytokines, people dealing with also other group doing metabolomics, but using MS. And we are doing lipidomics, and others doing proteomics, so that we are not sure. And we'll have access to the data, even if we don't do it by ourselves, but we'll have access to the inflammatory marks, but we are not, yes. That's the thing, we have, there are many intersections among those projects, of course, because we want to get the most out of these precious samples, right? Yes, and since they were kind of, it is the center, the NEG, right? From the university, it's for the community to use, but there are kind of different projects that you have to see what other people are doing. Yes. Right?
Yeah.
Yes. Yes.
Thank you for the presentation. It was great. And it's just a curiosity, actually. You showed in the introduction that we had the higher mortality in pregnancy, but it's not the same mortality that we have in everybody else in Rio, because we had higher mortality.
General high
Right.
mortality for pregnancy.
It was not exclusively for pregnant and COVID, right?
Yeah, we have a high mortality, but this was really increased into the COVID. But we already have.
Yeah, but no, in the rest of the population in Rio, we also had higher mortality.
The second highest
Yeah. Yeah. But doesn't matter if we're pregnant or not. It was even higher. Work. Yeah.
Because of at the beginning, the trial, the surveillance was very poor. So
Yeah, yeah. They didn't have anything in the maternity. Yeah. Yeah, it was very hard to collect tissue and information because of the everything. Thank you.
Thanks.
Mais alguém? Anyone else? Curiosity? Yes.
Hi, very nice talk. I was curious about the tryptophan data. That is, I think it's not related to glucose metabolism or diabetes, as you said. But do you have any experimental model to test what is happening there? Because tryptophan is precursor for serotonin, melatonin, and NAD too. So, and liver is the main organ that produces NAD from tryptophan. So maybe it's related to liver damage, or maybe it's related to a transport of tryptophan to the fetus, as you said, I think. So do you think about studying further what you saw with tryptophan metabolism?
So right now, I do agree with what you said. It's really hard to say that these increased or decreased levels of any metabolites is for more utilization, or if the organ that metabolizes it, it's damaged. But it seems that tryptophan plays a major role in this kynurenine and producing IDO and indole, I don't recall the name, but it's related to the inflammation. And we don't know if the tryptophan is lower because we have a major liver damage.
It's higher.
Sorry, if the tryptophan is high because you have a major liver damage, or if they are just, I don't know, the metabolic cycle is just speed up. And it's probably both, like the Jorge showed us in the presentation, the orchestra. I think it's everything happening at once. And right now, not on these samples, but we can rely on other studies that use models like mice, because it's easiest. But as I said, our previous study showed that tryptophan presented really decreased. And these in pregnancy showed a slight increase. So I don't know, probably we should get any other metabolites from the kynurenine pathway who can show us to what is really happening to tryptophan.
Okay, thank you.
Thank you.
Thank you, Juliana. I think studying tryptophan in these scenarios just of kind of a major importance, because a decrease in tryptophan is a marker of severe COVID. And what we saw was, the opposite, right? So placenta probably is playing a major kind of either serotonin or the I always forget the name of this metabolite, but also the using NMR, the sensitivity to detect tryptophan is low, kind of because the amount. So we are. So I think that we will do is to use a mass to not only to confirm tryptophan levels, but also serotonin, which is even lower the level. So that's one. But using cell culture model, we wish, but I don't know, maybe.
The discussions, I mean, that's fantastic discussion. And I was just said to Enrico, there's one of the papers there was one of the papers from my group around, I think this idea that the placenta is involved in serotonin is a really important one. And it gets back to the orchestra. The placenta is so important in, it's trying to mount some kind of anti-inflammatory response here. And I think, that's really key. The other thing that we published, I'm trying to recall the exact details, but it was a few years ago, was looking at placenta and the whole tryptophan pathways, and there was a big impact of age. So you might want to look at age in the context of these data sets.
So another thing that came up to my attention is because some pregnancies, you may have COVID being transferred to the fetus, and this is probably a placental misfunction or susceptibility or something that may make it happen. So I was wondering if maybe in your data, if you could go back and try to get the placenta and do some histopathological analysis. No, if you may have it, you know, because why do some worst cases have this profile, this metabolic profile? And it's the same questions. Why do some pregnancies you may have transfer of the virus to the mother? This is unsolved. Perhaps in your data, if you can dig in a little bit more, you can come closer to answer this question.
Of course, if we were capable of, we are collecting every tissue and about half a liter of blood just to keep some of them in the -20.
But so we want to be able to start a new cohort and to collect the placenta, because I think there's going to be a collaboration between the City Hall and the Secretaria Municipal de Saúde with the center here. So, and then we'll be able to actually, so we want these women and to follow not only kind of the one time, right, but also to follow and to get where they have the prenatal care and also delivery. So probably we'll be able. Yeah, so that. Yes, exactly. If there is a stimuli for these women to come here, even to test, even if they are, of course, with no symptoms, we'll be able to get more information of what the control group. Yes, amazing. Okay, thank you, Marcus.
Thanks for your attention.
Yes, thank you. And now moving on to the last talk of this morning. Isis Trevenzoli, thank you so much. Isis from the Institute of Biophysics. If you want, you can come. Institute of Biophysics from this institution. Yes, clever. And she's an associate professor there. And she's actually, the focus of her research is developmental origins, health and disease, mainly maternal diet, obesity, right? And study several aspects of offspring metabolic health. And she's really interested in the endocannabinoid system. That's what she's going to talk about. Isis recently got a very important award from FAPERJ, from Women in Science from the state of Rio de Janeiro, with this project. And I wanted to, with the cannabinoid metabolism and study. And I want to mention that she's been dedicated kind of really to implement a method for the analysis of these metabolites here. And hopefully in the near future, it will be a multi-user facility. And then that will be able to, we feel that when we are able to implement the method that we really learn. And yes, and I really congratulate you and your group for that. And thanks so much for accepting. And also the personal thing, she has 14 children, one human, two dogs, one guinea pig, and 10 fishes, right? And she also loves to cook and carnival and beach and sun. Yes, so thanks so much.
Hello, everyone. Good morning. I will try to lead with 2 pointers and this mic and my time. Okay, so today I'm going to talk to you about this project that we developed in the Laboratory of Molecular Endocrinology. And the title of this presentation will be Maternal Nutrition and Offspring Development, and how the cannabinoid system is related to the programming induced by maternal nutritional insults. Let's see. Okay, so today we are going to talk about, very briefly, about the developmental origins of metabolic diseases and how the endocannabinoid system may be an important role in this context. And I'm going to present some new data we obtained in the lab. Some of these data were obtained in an animal model, rat model of maternal obesity induced by high-fat diet. And I'm going to show you some data on liver endocannabinoid system in the offspring. And lastly, not less important, I'm going to show you some recent data in the MILCA project, in which we are investigating cannabinoids in human milk. So most of you, I believe, it's familiar with this hypothesis that is called Doherty hypothesis, in which we know that hormonal, nutritional, or environmental influences during critical periods of life, such as gestation, lactation, up to 2 years of age of the offspring, may modulate epigenetic profile of the developing fetuses and the developing child, and increase the susceptibility to develop metabolic diseases such as obesity, diabetes, hypertension, and non-alcoholic fatty liver disease. And in our lab, we are particularly interested in how maternal high-fat diet or maternal obesity can increase the susceptibility to develop metabolic diseases in the offspring. And one of the systems we have been interested in, I guess, in the last 10 years maybe already, is the endocannabinoid system, which is a physiological system that controls homeostasis in basically all of our tissues. And it's a lip signaling system. And the ligands, the classic endocannabinoids, such as anandamide and 2-arachidonylglycerol, are bioactive lipids derived from membrane phospholipids, and these endocannabinoids, and also the most known exogenous cannabinoids, THC, binds to mainly to 2 receptors, G-coupled protein receptors, CB1 and CB2, cannabinoid receptor type 1 and type 2, but also other receptors such as GPR55, TRPV1, and others, to act. And they act very rapidly, and they are produced on demand in all of our tissues. And after action, they are metabolized by several specific enzymes, such as the fatty acid hydrolase enzyme that metabolizes mainly anandamide, and the monoacylglycerol lipase that metabolizes mainly 2-arachidonylglycerol, which I will refer as 2-AG. And importantly, diet, it's a major controller of this system, because we have to think that these molecules, they are lipid derived. So they are basically derived from arachidonic acid, contained in the phospholipid membranes. So diet can, especially dietary lipids, can strongly influence the levels of the lipid ligands and also the enzymes and the cannabinoid receptors. So because this system is a lipid signaling, it's present in all tissue, and apparently it has an important role in several early life mechanisms. We thought that maybe would be a role for the system in the context of developmental origins of health and disease. For example, the endocannabinoid system controls mechanisms since very early stages of life, such as blastocyst implantation, placentation, neurodevelopment during gestation, and across lactation, it's important to suckling initiation and feeding, and also development of adipose tissue, white and brown adipose tissue, and continues being important for the neuronal development. In adolescence, it has a special role on the development of food and drug addiction and stress response, and all of these, if we have changes in this process across life, we are more susceptible to develop metabolic and mental health issues during adult life that may be involved in this regulation of the endocannabinoid system. So particularly regarding energy metabolism, the endocannabinoid system, when it's activated, it increases appetite, increases food intake, and also the preference for lipids in the diet. And peripherally, it increases visceral fat, lipogenesis, both in adipose tissue and liver, and also increases glucose uptake. And interestingly, several studies have been shown that polymorphisms in the genes coding for several components of the endocannabinoid system are also related to metabolic dysfunctions. So in our lab, we studied a rat model of maternal high-fat diet. And in this model, we have been investigating the endocannabinoid system in several tissues of the offspring in several time points of life. And here is it's a picture of we have until now for the phenotype of these offspring. So they develop early obesity, dyslipidemia, and they present not only increased food intake, but also a preference for fat since adolescence. And they also have central leptin resistance at the hypothalamus level. And we also characterized recently that they develop anxiety-like behavior since adolescence. And mostly of these changes were associated with an increase of the tonus of the endocannabinoid system in the offspring in the brain adipose axis. So a few years ago, maybe 3, I guess, we asked if there would be some changes occurring in the liver of this offspring, because we know that the liver is a central organ on lipid metabolism. So this paper was, the results I'm going to show you on this paper and the next few results were performed by Larissa Fassarella, which is around here, she's there, which is a master's student in the lab. So at this time, she was an undergrad student, and also Rosiane Miranda, at this time she was a postdoc at the lab, and now she's already assistant professor in another university here in Rio. So in this paper, we showed that this model in maternal high-fat diet model in the liver of the adult offspring when they were 6 months old, they present in the liver increased expression of CB1, CB2, and also the metabolizing enzymes of the endocannabinoid system. And this occurs only in male offspring. We did not observe any change at this age in the liver of female offspring. And these changes were associated with hypertriglyceridemia and liver steatosis, also only in the male offspring. In both male and female offspring, we observed oxidative stress markers increased in the liver and also associated with mitochondrial morphological changes in this offspring. Therefore, because we only saw the changes in the endocannabinoid system in male offspring in this context, we speculate that this system is more related to the regulation of lipid metabolism rather than other oxy damage and mitochondrial damage that we observed in both sexes. And then we question, how about the timing? Because this previous study was conducted with the offspring in adulthood. So we wanted to know what the time these changes in the endocannabinoid system were occurring at first. And we also asked if it would be possible to prevent these changes by maternal fish oil supplementation. And why fish oil? Because the endocannabinoids, anandamide and 2-AG, they are derived from arachidonic acid and a 6 fatty acid. And there are lots of publications in the literature showing that fish oil, which is rich in any 3 fatty acids, could balance and re-equilibrate the levels of the endocannabinoids. But nobody had been shown this in a metabolic programming model. And this project is being developed by Larissa. It's a continue of her work, and now she's a master student, and also by Jessica Jezebel, postdoc at the lab. So here is the design of our animal model. Let me check my time. Okay. And in this model, we divided adult female rats into 2 groups initially, a control group that receive diet containing 10% of fat, and a high-fat group that receive a high-fat diet with 29% of fat. And the rats receive this diet 8 weeks before mating, during all gestation, and during all lactation. And after weaning, the offspring receives only control diet. So the nutritional manipulation is concentrated in these critical periods of life. And in this project, we included a 3rd group, that is here, in which we supplemented the pregnant rats with 3% fish oil in the high-fat diet. So we have been following this offspring in several time points of life, and we have collected some data on the adult life, as I just showed you, but we don't have data still on the adult life of offspring whose mothers received fish oil supplementation. This is under construction, and also the data on offspring at weaning is also under construction. And today I'm going to show you some of the data on the liver of the neonatal offspring of these three groups. Control group, which I will refer as CT group, high-fat group, and high-fat fish oil group. And we always analyze males and females separately. Okay, so we first look at the content of triglycerides in the liver of this offspring, neonate offspring, and we did in the male offspring, we did not find any difference between among the three groups. The control group, maternal high-fat diet and maternal high-fat diet fish oil group. But when we look at the genes coding for the enzymes involved in lipogenesis in the liver and also lipid oxidation, we already see some modulation of gene expression before the phenotype comes in. So we observed a profile of increased changes in genes related to lipogenesis with the high-fat diet, and a rescue profile when the mothers were supplemented with fish oil. And we also see a profile of mRNA changes regarding lipid oxidation, in which maternal high-fat diet decreases, for example, PPAR alpha and CPT1 beta, and the fish oil, but the fish oil did not change this profile. And regarding the female data, the liver of female neonate offspring was not affected in terms of lipid content in the liver, triglycerides content in the liver, but the fish oil supplement already induced an effect of lipid reduction in this liver. And this was associated with, again, modulation of genes coding for lipogenesis, but not here in females, not modulation of lipid oxidation markers. So we next went to investigate the endocannabinoid system in this offspring. And we observed that in males, we have an increase of CB1 content in the liver of the offspring, HF offspring, and this profile was rescued by the supplementation of fish oil in the mothers. We didn't see any changes in CB2, but we didn't see significant changes in the levels of liver, anandamide and liver 2AG with the maternal high-fat diet. But there is a trend, but the fish oil supplementation reduced these ligands in the liver of this offspring as expected. So these results indicate that in maternal high-fat diet offspring, there is an increase in CB1 signaling in the liver, and that maternal fish oil supplementation can rescue, at least partially, this dysregulation of the endocannabinoid system in the neonates. So this is important, I guess, because it has been shown that CB1, the stimulation of CB1 in the liver is associated with the development of hepatic steatosis, because it is controlling the genes related to fatty acid oxidation and VLDL clearance, and it's also involved in hepatic fibrogenesis and also apoptosis in other models of liver injury. And regarding the female offspring, we did not observe changes in the CB1 content induced by maternal high-fat diet, but we did see an important reduction of CB2 content of these receptors in the female offspring. And also, we did see a decrease in the ligands, anandamide and 2-AG, in the offspring with the fish oil supplementation. So these results indicate that we have in liver of female offspring a decreased CB2 signaling, and that, again, that the maternal fish oil supplementation can decrease the levels of the endocannabinoid system in the liver of neonate rats. So, and this is important because CB2 is associated with an anti-inflammatory profile, and decreasing CB2 in the liver of the female offspring may be detrimental because it may contribute to increase inflammatory profile in this tissue, which may have some metabolic impact in the long term in this offspring. So when we have activation of CB2, we have decreased the inflammation in the liver, decreased the steatosis, and decreased apoptosis. And because the females have less CB2, they are less protected. So for this experimental part, we can conclude so far that maternal high-fat diet modulate the liver endocannabinoid system of the offspring before obesity and steatosis development. That maternal fish oil supplementation during pregnancy may reprogram the endocannabinoid system and liver lipid metabolism of the offspring. And we wanted to go for future studies that we could address the long-term effects of maternal fish oil supplementation on offspring health. And now I would like to share with you some data on the MILK project, the MILK project, in which we investigate the cannabinoids in human milk samples. So the milk, the breast milk, it's a very complex biological matrix. And this composition may vary according to some metabolic and anthropometric characteristics of the mother. And here you can see the general composition of human milk, basically lots of water, but also at least 4% of fat, considering a milk from eutrophic women. And also, we have, of course, carbohydrates and proteins, and a smaller amount of hormones, growth factors, microbiome, cytokines, microRNAs, and etc. And we were interested in how maternal nutrition and maternal health could impact the endocannabinoids, the levels of endocannabinoids in this complex matrix. So for that, we recruited almost 150 women, but from those women, we included 92 we had some losses, as you can see here. And this study was conducted by Tatiana Fernandes, a master's student. She just concluded this year, a master's student in the physiology program, and also Ana Lorena Ferreira, a postdoc from the Nutrition Institute of our university in collaboration with Dr. Gilberto Cacchi. And these women, from these women, we collected milk samples in 3 time points. And we collected at time 1 from samples from women between 2 and 8 days postpartum. And we collected 41 samples at this time point. The second time point, before that, this time point is important to emphasize that we are collecting mostly colostrum, okay? And in the second time point, where we do have mature milk, we collected 67 samples between 20, 28, and 50 days after birth, and the third time point at 3 months after birth. And these women were stratified by pregestational BMI, under trophic or overweight or obese women, and also were stratified by their gestational weight gain in excessive or non-excessive gestational weight gain. So we first wanted to look if there was a variation in the levels of the 2 major endocannabinoids, anandamide and 2-AG, across these 3 time points of lactation in the milk. And we did see a robust variation in both anandamide and 2-AG among the time points evaluated. So for anandamide, we see a robust increase from colostrum to 1 month milk, and then there is a drop to the levels of the colostrum in the milk. For 2-AG, we also see a dramatic increase from colostrum to mature milk, but then it remains with high levels in the next time point at 3 months. So we have a drop in anandamide, but the levels of 2-AG remains high from T2 to T3. So we are still thinking about the physiological relevance of these changes, but because of our knowledge about the physiological role of the system in general, we speculate that these changes, this fluctuation may be related to suckling initiation in this rise over here, neurodevelopment, because endocannabinoids are very important neuromodulators, feeding stimulation, and also, more recently, we are thinking about sleeping regulation. And when we stratify these samples by pre-pregnancy BMI and gestational weight gain, we saw that women with obesity or overweight present increased levels of anandamide in mature milk, T2, and an excessive gestational weight gain was associated with increased levels, or almost significant increased levels of milk 2-AG also in T2. So here we are talking about mature milk. We didn't see any changes in colostrum. We evaluated several parameters, and it seems that in the colostrum, everything goes right. And after that, that first week, that change started happening. So we also evaluated, besides endocannabinoids, we also evaluated important hormones for metabolism, such as insulin and leptin in the milk, and also the content of proteins and lipids. So here I'm showing to you a Pearson correlation plot, where we plot these two parameters of the mothers, pre-pregnancy BMI and gestational weight gain. And here several milk components at T1, T2, and T3 time points. And every time you see a circle, it seems that this correlation, it shows that this correlation is significant. And as darker and as bigger is the circle, more stronger is this correlation. So I wanted to just to point out a few things here. And the first thing is that the pre-pregnancy BMI was correlated with increased levels of leptin in mature milk at T2 and T3. And this was expected from the literature. And also that the leptin was positively correlated with the levels of anandamide in this milk. And lastly, before that, we saw a very strong correlation between gestational weight gain and the levels of 2AG in the milk of 3 months. And also we saw a strong correlation between the presence of the endocannabinoids in the milk in the 3 time points and the levels of triglycerides in this milk, which was kind of expected because the higher is the lipid content, we expected to have a higher, we hypothesized that we would have a higher levels of these bioactive lipids. So from this human cohort, we can conclude so far that the milk levels of endocannabinoids change across lactation, with anandamide and 2AG present different profiles, that pregestational maternal obesity or excessive gestational weight gain are associated with higher levels of endocannabinoids in human milk. And we know that more students are needed to identify the precise physiological role of these milk endocannabinoids and their contribution to the development of origins of metabolic diseases. With that, I'd like to say thank you to the students that work hard at the lab and that are helping me to construct this knowledge and this history, and also some of the collaborators, and once again the organizers Tatiana and Tanya for inviting me to share some thoughts. Thank you.
Thank you, Isis. Questions?
Hello, Isis. We have our desks just next to the other one. I have never seen these results. That's very good that we have the workshop.
They are brand new, I have to say.
So I look at the graphs that you show that the levels they go up with the gestational gain weight, but you have a lot of variation. Did you see if this variation, they go up with the amount of gain weight, like or the BMI? So more weight they have, more they have the
not the BMI, but the gestational weight gain actually. Yes, I think it is, and the correlation plot shows that we have a very strong correlation in this data that is not stratified
Right.
in two groups. So we consider all the groups, all the women, and we do see a strong correlation with the gestational weight gain.
And another question, in the breast milk, you have circadian variation? Because you talk about the sleep.
Yes, this is very interesting. We just started a project to study this circadian rhythm of the endocannabinoids in obese people. And there is one paper showing that there is a circadian rhythm in the milk where the levels during the day are higher compared to the night levels.
And from the bank that you use the milk, do you know what time they collect the milk?
Yes, they all were collected in the morning. So we
You can
cannot,
see.
we cannot see it in this cohort, but we have these in mind. And Alberto, which is a new PhD student, will do a study on this in obese people, not pregnant, but first in obese people.
Very interesting. Thank you.
Thanks. That was really fascinating. As you know, in North America, the recreational use of cannabis is now legalized. And so I'm wondering what that might look like layered onto the top of this fascinating sort of story that you've just presented would mean, because certainly as someone who lives now in Toronto and is exposed to some of the recreational activities, it's actually difficult to go very far in Toronto without smelling quite significant amounts. So there's a lot of secondhand smoke that's around as well. So right outside actually our maternity hospital. So I'm just wondering what your thoughts around the implications of that might be.
Yeah, this is the fact that I just came from Toronto and from a meeting on endocannabinoids there, and and this is a reality in Canada and in North America in general. So we have studies on the effect of THC and also cannabis exposure in pregnant women. And in general, it's very similar to tobacco maternal smoking exposure. It compromises placenta function. It compromises fetus growth, induces fetal growth restriction. And there are some data from Spain showing that the specifically exposure to THC, isolated THC in some time points during gestation, I guess in the day 17 in mice, it results in several cognitive impairment and behavior problems in animals. But about milk, I've never seen data on milk, actually. I don't know if there is something out there, but I've never seen. But I think it will be an impact. That we know that in non-pregnant people, the exposure to cannabis alters the levels of CB1 in the brain, and this has been shown for humans, indeed. But we don't have studies on that. It would be very interesting. But in Brazil, it's very difficult to conduct this type of studies because of legal issues.
Thank you.
Yes, thank you so much for your presentation. It is more of a brainstorm than anything. But when I speak with developmental immunologies, they always claim that the liver is more of a immunological organ during pregnancy rather than a metabolic one. And very little is known as to when the liver, transition to more of metabolic organ, right? So I was wondering or thinking whether your change in the maternal milk, in the milk, wouldn't be associated with this liver transition from immunological organ to a more metabolic organ during, early stages of development post birth.
It's an interesting thought. But one thing to consider is that the endocannabinoid system plays roles in both in metabolic and inflammatory profile. So I think it's difficult to dissect because it's really playing the game in the two in the two sides. But it's an interesting to thought about it. Yeah. Thank you.
Hi, this was very interesting. I'm not really familiarized with the endocannabinoid system as much, but as you mentioned, it seems to be related to maternal diet as a source, as a precursor of the endocannabinoids. So I was interested to know, like you mentioned the high-fat diet, and like was the lipid source something that you accounted for? So usually it's high fat and it's higher in saturated fat, but how about the arachidonic acid or even linoleic acid? Is it different from the control diet, and could it be part of the explanation? And also for the milk project, I was interested to know where it comes from. So, because we know that sometimes lipids in milk, they come from de novo synthesis in the mammary gland. So, where is the endocannabinoid system playing a part in all of this? And could it, be something to explore?
Okay. First question was related to dietary lipids, right?
Yeah, in the high-fat diet.
In the high-fat diet, yes. In the animal model, we have these experimental diets analyzed, and it's rich in saturated fatty acids. It has an increased ratio N6 and 3. And we think that this is the profile leading the changes in the endocannabinoid system we are seeing in the offspring. And these changes we observed in the liver, but also in several other organs in different ages. In general, the endocannabinoid system is upregulated in the model in several tissues, in the brain, including the brain. And we think it's related to that profile of the diet. And for the women cohort, we did analyze the diet profile of these women. I did not show the data because it was not significant, but we did not see a strong correlation between the dietary lipids, and we analyzed like arachidonic acid and other N6 and 3 and saturated fatty acids and the total lipid content of the diet, and it was not significant correlated with the endocannabinoids in the milk. But we have a limitation on this data because the milk was collected in these 3 time points, and the interval, the frequency food frequency questionnaire in the mothers was performed in their 3rd trimester. So there is a distance between the time of we question about the diet and when we actually measure the levels of the endocannabinoids. But with this design, we didn't see strong correlations. And the third question is where it comes from, right? So we don't know exactly. What we do know is that in mammary gland, at least in the experimental model, we have detected the enzymes that synthesize both endocannabinoids. So we know from the animal model that the enzymes are there. And what we actually think is that it's coming like leptin, half and half. Half from maternal circulation and maternal lipid storages like liver and adipose tissue, and another half from diet. This is what we speculate, but in fact, we did not have data on the literature to confirm this speculation, okay?
Okay, thank you.
Thank you.
No, I just have one, maybe not a question, but a comment. It seems, thank you so much for your lovely presentation, for accepting our invitation, of course. It seems that it definitely related to adiposity, right? So one thing, because you see an association with higher pregestational BMI, higher the level of, one of the two, right? Cannabinoids. And also gestational weight gain, as in Tanya's question. Maybe we know it is well described that the higher the pregestational BMI, less weight gain. So you can associate, probably those are different women, if there is also a matter of adiposity, right? And maybe it is regulating adiposity in the infant. Do you have data on the adiposity on the newborn? But maybe in your animal models, because it's...
Yes, two things. This weight gestational gain was, this stratification was based on the pre-pregnancy BMI. It is already adjusted for that, and we don't have the data on the babies. We have all the data. We have Dr. Kak's group have data on microbiota. They have data on cognitive function of the infants up to 2 years, and inflammatory data and other things in the milk. Yeah, we don't have adiposity in this study, and there is no paper in the literature showing this association. There are papers showing the correlation, the positive correlation between milk levels of leptin and adiposity of the infants up to 4 years of age. But about the endocannabinoids, we have nothing. And from the animal models, from our animal model, it's very difficult to discuss the milk levels of endocannabinoids because in the animal model, we found the contrary. and it's dramatically reduced in the milk of rats. And on the other side, we think that we have an adaptation occurring in the offspring that may be a short-term good adaptation, that the offspring increases the levels of the receptors everywhere to at least guarantee early development at certain level. But in the long term, this becomes an disadvantage because it makes it increases food intake and food preference for fat and obesogenic diet. So in the animal model, is still we have a different profile.
Thank you. Yes. Well, now we finished the morning session, and we can come back. Tanya, as we keep to the schedule, 1:30. What do you say, 1 hour? Yes. Okay, yeah, a quarter to 2. 1:45, yes. And we on the third floor, so we can all go. Hopefully it will all good. So, a janela virtual no terceiro andar, gente, se vocês continuarem.
Não sei nada. A minha não. I have a pen drive. Steven, Enrico, do you want to see the mouse? I can show you how to use. Because sobe pelo. Não, é um mouse. Você tem que apontar para o computador, você não pode apontar para a tela. Eu dou aula com ele, eu tenho eu dou aula com ele o tempo todo. É só uma questão de costume, que normalmente meu computador está lá. Aí eu apontaria assim. Funciona para lá. It's a Bluetooth mouse. So the Bluetooth is there.
Can't do my head in.
It's not impossible.
Here, I've got a better idea.
It's not.
I can't do that. I'll get it
Of
all.
course you can do. You can't use another pointer because then you have to change. You go. You control here method powers, and then. Põe o slide para mim de novo, a apresentação de alguém. Isso, aqui. Sim, Steve. A Tânia.
Which one do I press?
2 points. And then...
Yeah, I know, but I can't. I'm pointing at the screen.
Vamos lá, eu vou explicar.
É o mouse, eu tenho
Não, mas a dificuldade está acontecendo quando você bota para a direita, você pode voltar para a esquerda.
Go to the left side.
Deixa apertado para a esquerda, totalmente. Leva mais. Isso. Ele está lá na minha tela. Vai lá, vai lá. Então a pessoa, ela aperta e está lá na minha tela. É simplesmente puxar para a direita.
O pointer normalmente, ele aparece quando você aponta. Só que como ele é um mouse, você tem que fazer ele aparecer. Eu dou aula com ele. Eu uso exatamente isso. Só que normalmente eu ponho, porque ele funciona na TV, né? Então eu ponho o computador sempre na minha frente e aí eu já consigo. Tá, agora não posso acostumar.
Vou colocar ali.
Você pode apontar. Você pode apontar para a tela.
Eu tenho que apontar mais para lá.
Você tem que fazer ele aparecer. Tá? É só você fazer ele aparecer. Não interessa onde está a sua mão. Quando ele aparecer na tela.
Entendi.
Aqui é simplesmente a segunda tela. E está na esquerda, ó. Está lá na esquerda. Está lá na minha tela, lá em cima. Você puxa ele para a direita.
Mas assim eu tô conseguindo.
Entendeu?
É só ir para a direita.
Isso. Exatamente. Se ele sumiu, você quer dizer que ele está lá. Eu estou vendo ele, não é você.
Pode.
Entendeu? Porque aqui é só você imaginar que aqui tem um outro monitor aqui. Entendeu? Tá.
Aí você aponta no. É isso aí. Tá bom? Obrigado.
Obrigada, Vinícius. Hello, welcome back. Can we start? Podemos começar? Because we are, not that we are on time, but Brazilians are. No, so that I was going to say, I was going to be ironic. Brazilians really, yeah.
I'm very tight with my time.
Yeah, I try to be, yes. So, people, thanks for staying here, continuing here, right? Of course, and it's my pleasure to introduce Tanya. So she's going to be the first speaker of this afternoon session. Not only she's one of the organizers, and she the idea, the concept, right? The embryos came from her, of course, in 2017, right, Tanya? That you had Enrico and Steve Gates, how to say, funding, and so we've been together since 2017. And Tanya is a full professor at the Institute of Biophysics. She works with different aspects of the physiology of reproduction, including thyroid hormones and vitamin D. That's what she's going to present. Tanya is a renowned scientist and researcher in the endocrinology field. She has a vast experience in editorial for major high-impact journals in this area. Tanya at the Institute of Biophysics was a coordinator of the postgraduate program and also outreach program. So many activities which are kind of time-demanding and very important. And also for the personal side, she's how to say, she loves pets. Yeah. She's always with a pet lover. She's always a pet. She has a dog.
I have 2 dogs.
Birds.
I have 2 dogs, 2 birds, some fish.
Yes.
Yeah.
So the same as as Isis.
Right.
Okay.
Right.
Okay.
So my dog is Isis' mother's dog.
Okay. Yes.
Yeah, she is a very nice dog with 17 years and 3 months.
17.
17.
Dachshund.
Yeah.
Okay.
I think it's the only one that survives that long.
That long. Okay. And also, just to finish, she's a exercise holic, so adrenaline rush. I think maybe in this, presentation. Thanks so much, Tanya. Yes.
Okay, thank you, Tatiana, and thank you everybody for being here in this nice weather. So it's actually good to have a place with air condition for the full day, and we don't even have to go outside to have lunch. So this is good. And I actually, I'm bringing you one project that I plan to say that it was new in the lab because I just have preliminary data, but I was thinking yesterday, and I was actually writing the project when Enrico arrived in the lab in 2012, and I was supposed to start the vitamin project like 11 years ago, and then when the Gates grant arrived, we had to stop everything, and the vitamin D just stayed there in the corner waiting for we to accomplish so many things with the Gates grant, and then now we are back to the vitamin D. So as I told you, it's preliminary results, and I brought some facts in the literature to show you what we know about vitamin D and the reproductive system. And I remember that when I planned the project, when I started thinking about the project in 2012, they had one review that tried to cover everything that we knew about vitamin D and the reproductive system. And after reading, I just conclude that everything was gossip. We actually didn't have enough data, and we still don't have enough data in humans to be sure about all the roles that possible actions of vitamin D in the reproductive system. So I'm going to show data from the literature about vitamin D and the reproductive system, a little bit of vitamin D and fertility, and then at the end, I'm going to show our preliminary data about vitamin D and pregnancy in one animal model. So as all of you know, to have the active vitamin D that it's produced by the kidney, we need to get the precursor of vitamin D from diet or from the sunlight. And vitamin D deficiency is one of the most common medical conditions worldwide. And in 2007, we had an estimation of 1 billion people in the world with vitamin deficiency or insufficient. So several effects of vitamin D that are very well established, like the bone turnover, and also the effect of vitamin D in the intestine to increase calcium absorption. But several other non-classical effects have been studied for the last 2 or 3 decades. We have very well-established non-classical effects in the immune system. And as you can see here, the researchers are exploring actions of vitamin D in the placenta, ovary, that includes steroidogenesis and folliculogenesis, endometrial, implantation and endometriosis, and also in a male reproductive system in the testes. So we have a good amount of information about the association between vitamin D, fertility, and semen quality in men. Here is a systematic review that reviewed a good number of studies and showed that vitamin D serum levels were higher in fertile men compared to infertile men. They also showed that data in the literature confirm that sperm motility was increased in men with higher levels of vitamin D. So when you look at the male reproductive system, you have a good amount of data in human that you can confirm the effects. Here is one paper using an animal model, a rat with vitamin D deficient, where they showed less proliferation and greater apoptosis of testicular germ cells. When you go to the female reproductive system or pregnancy, then we have lots of papers that are very hard to evaluate and actually confirm the effects. So in this paper here, that it's a review, they showed several factors, biological, social, and behavior, that can affect the vitamin D levels in a pregnant woman. So one of them or 2 of them are obesity or weight gain during the pregnancy. So because of the lipids nature of vitamin D, the adipose tissue can actually store vitamin D, and then the levels are decreased. Of course, adolescence pregnancy is another risk for low vitamin D levels. We have social factors like living in higher latitudes than 40 degrees, south or north, doesn't matter. The fact that you live in the city or in the countryside can affect your vitamin D levels. Social class, adolescence pregnancy, again, ethnic origin because of the amount of melanin we have in the skin, outdoor, the amount of outdoor physical activity, diet, insufficient vitamin D. Some places or some countries, they add vitamin D in food. We have the use of vitamin D supplements that in some countries is very popular, and vitamin D is a hormone, so we should not use as a supplement without medical encouragement. And also we have behavioral factors like the tendency to avoid the sun. Here I can add the pandemic and all the time that people had to remain indoors in big cities, for example. The use of winter clothes or clothes that cover the whole body for different issues like religion or climate, and the use of sunscreen. And this review highlights that there are no studies in the southern hemisphere that assess vitamin D status through the life course of the woman. So most of the studies are in the northern hemisphere. Because of vitamin D has lots of effects in immune system, we have lots of papers that looked for an association between vitamin D and endometriosis, but in the systematic reviews that the last one that was from 2019, they could find any association between vitamin D deficient and endometriosis. However, when they look at the polycystic ovary syndrome patients, they have this interesting paper where they use, that it's a randomized, placebo-controlled clinical trial, where they used vitamin D and calcium together with the metformin treatment to regulate menstrual cycle abnormalities and the hormonal profile of these women, and they conclude that vitamin D supplementation would help these women to normalize the menstrual cycle. And in this systematic review, they study populations that were going through in vitro fertilization, and they looked at vitamin D deficient is associated with a poor ovarian stimulation outcome. For people that are not used to in vitro fertilization, usually women with a polycystic ovary syndrome, they respond very well to ovarian stimulation. However, if they are deficient, vitamin D deficient, this response is not really good. We have a project in a collaboration with private clinic of human reproduction, and we try to collect the same data to show an association between vitamin D and several parameters of the in vitro fertilization, but we could not see any important correlation. So here you can see the number of the retrieved oocytes, and they don't get better or worse depending on the vitamin D levels of the patients. I brought this paper that is very interesting because it is the vitamin D deficient associated factors among pregnant women in a sunny city of the northeast of Brazil. And I think that it's fair to say that Rio is an example of a sunny city in the southwest of Brazil. So we can actually look at this data very closely. And they show that even in one city that has sun most of the days of the year, the levels of vitamin D decrease from summer to winter. And we are talking about pregnant women. And in this population, they found that 40% or 45% of pregnant women present vitamin D insufficiency, 24% vitamin D deficient, and 31% of vitamin D sufficiency. And this is something that I'm always very curious about, how we get so much sun and we can't actually have most of the population with vitamin D sufficiency. And the authors compared this data with other population, and they show that this is the same picture that they find in Spain, for example, but it's much better than when they compare to the Norway population, for example, where they have much less sun during the year. So actually, to maintain the vitamin D sufficiency in pregnant women, it looks like it's harder than in non-pregnant women. However, all the human studies present limitations. So we have heterogeneity between studies. So they have different study populations, ethnicity, sun exposure, and season. Most of the human studies, they are cross-sectional studies. Therefore, it's impossible to draw a causal link between the variables. Also, most of the human studies ignores the polymorphisms that are very frequent in vitamin D receptor, the VDR, and also the vitamin D binding protein. Therefore, all the systematic reviews that deal with vitamin D in humans, they always say that we need prospective cohorts. I was doing really well with the pointer. I'm going to get back to it. So we all agree that we need prospective cohort, more clinical trials and preclinical studies to actually formulate better the vitamin D importance in the reproductive system. Well, this is one paper from, I think, 2010 or 2011. I cut the reference, but it's about that time, where they describe a knockout mouse that that does not express the VDR, so does not respond to vitamin D, and they showed in the phenotype uterine hypoplasia and, of course, growth retardation. So if you look here in the knockout mice, they have an important uterine hypoplasia, and the uter responds to estradiol treatment, as you can see here, but even after the treatment, it does not reach the normal size in the white type compared to the white type mice. And here is the ovary. So the ovary of a white type with a nice follicle, and the ovary of the knockout where you cannot see really the development of the follicles. Well, so in our lab, we are exploring the ovary and the placenta. So the ovary, we are doing in vivo and in vitro experiments, and we have a group of students that are working with vitamin D projects. So we work together with the other PI of the lab, Dr. Flavia Blois, and one master student that it's Marcela, and two PhD students, Hannah and Victoria. And for the animal models, we choose the mouse. So like humans, mouse produce vitamin D in the skin after being exposed to sunlight, and also through the diet. However, different from humans, we can control the amount of sun that the mice receive. So in the lab, they receive no sun at all. So if you control the diet, you can be comfortable that they are deficient or supplemented or normal vitamin D. So we choose the mouse model to develop the gestational model of vitamin D deficiency and sufficiency. So what we do, we use a controlled diet with different amounts of vitamin D. We have one deficient group with no vitamin D in the diet. We use what we consider the ideal amount of vitamin D, and I'm sorry, it's in Portuguese here, so it's at 1,000 international units of vitamin D in the control group, and 10,000 international units in the supplemented group. They receive the diet before mating, like 15 days before. Then after mating, we sacrifice the animals around 15 days after the matings. So as I told you, it's preliminary data, and most of the data that I'm showing here, it's from the PhD thesis of Victoria, but Victoria right now is in Cambridge working in Amanda's lab together with, I can't say his name in Jorge. I can say in Portuguese, not in Spanish. And the most important result I have here in this slide is the number of resorptions that you can see here in gray, that the deficient group has a significant percent of resorption increase compared to the control and the supplemented groups. This goes nicely with human data that in 2022, this systematic review showed an association between serum vitamin D levels and spontaneous abortion. Patients with recurrent spontaneous abortion had lower serum vitamin D levels than normal pregnant women. And they suggest that pregnant women with vitamin D deficiency might be at higher risk of recurrent spontaneous abortion. And I brought another paper to about the same idea that showed that the risk of preterm birth decreases with higher vitamin D levels. Of course, if you know about the levels of vitamin D, it's really high levels that actually show this decrease of the risk of preterm birth. We actually look at the fetal outcome, so you can see that the fetal weight and also the placenta weight are decreased in the group that was fed with the vitamin D deficient diet. And again, if you try to correlate with human results, you can see here in this systematic review that there is a consistent association between vitamin deficiency during pregnancy and an increased risk of low birth rate. Because of the size of the placenta, we look, of course, in the proliferation and the apoptosis rate in the placenta, and we saw that in the junctional zone, both deficient diet and the high vitamin D diet decrease the proliferation. This U response, where the deficiency and the high levels of vitamin D get the effects in the same direction. It's not rare in the vitamin D effect. So this is good to highlight the idea that we should not take vitamin D without any control. We could not find any changes in the apoptosis rate in the placenta. We looked in the calcium deposit, and this is a collaboration with Dr. Amanda Sferruzzi-Perry lab. So Victoria had a lot of help in the lab to get these results. And you can see here that the deficiency of vitamin D also decreases the amount of calcium deposit in the junctional zone of the placenta. Also in collaboration with Dr. Amanda's lab, we look at the iron deposit in the placenta. We know that there is a relation between anemia during pregnancy and deficiency of vitamin D. And you can see here that in the labyrinth zone, the deficiency of the diet with low vitamin D, of no vitamin D addition has really low levels of iron deposits, but also the supplemented group also had a decrease in the iron deposits. As I told you, it's very preliminary data. So we look at the expression of 1-alpha-hydroxylase expression for people that don't have this in mind. This is the enzyme that activates vitamin D in the kidney. And the placenta expressed this enzyme, so we look at the expression, and we saw an increase in the vitamin D treated group of the expression of this enzyme. We also look at gene expression of VDR, calbidin, I don't know if it's right in English, but it's the protein that binds to calcium, and it stimulates by vitamin D. And you can see that deficiency of vitamin D decreases the gene expression of VDR and the calbidin gene. We also look at the 24 alpha-hydroxylase. This is the enzyme that inactivates vitamin D, also in the kidney, but it's also present in the placenta, but we could not see any difference. We look at the VDR expression at protein levels in the placenta, and we saw an increase in the vitamin D treated mice in both regions of the placenta. So to finish the presentation, I think I showed you data from the literature that showed that vitamin D deficient affects male and female reproductive capabilities. The literature data also suggests that vitamin D supplementation can improve the reproductive response to metformin treatment in human with polycystic ovary syndrome. The preliminary data from our animal model showed that vitamin deficiency during pregnancy corroborates data in humans, showing that low birth weight and increased risk of miscarriage in vitamin D deficiency. Also, although our preclinical studies at early stage, we have shown that the placenta changes with vitamin deficient and excess. And regarding proliferation, iron deposition, and 1-alpha hydroxylase expression, both lack and excess of vitamin D affected the placenta in the same way, confirming that for many functions, the excess of vitamin D should be avoided, as well as the vitamin D deficiency. So just to finish my presentation, I have to first thank the FAPERJ, CAPES, and SINPQ that I forgot to add to the final slide. I want to thank everybody in the lab. They are not here. We have more people, but we are. This is the newest picture that were in the last meeting that we were together. I want to thank Dr. Flavia Blois, that it is the other PI in the lab, and of course she helps me with all the projects that we have right now. I want to thank from the University of Minas Gerais, Dr. Henrico Blois and Dr. Fernando Reis, that collaborate in many projects in the lab. I really thank everybody in Dr. Amanda Sferruzzi Perry lab in Cambridge. They were great to receive two of our PhD students there. And of course, I have to always thank Steve Matthews from the University of Toronto, because he has been here in all three workshops, and the first one was because of the beginning of collaboration between my lab, Henrico, Fernando, and Steve. So thank you very much for all your help. And I'm going to finish thanking Tatiana Elbacha from also the Federal University of Rio de Janeiro for organizing this wonderful workshop that I'm gonna talk more about the workshop at the end of the presentations. So thank you very much, and I'm open for questions.
Thank you, Tanya.
Yes, of course.
Thanks very much, Tanya. That was really interesting. It's something that I haven't really thought much about myself, vitamin D and placenta, but in terms of, you saw some, I mean, you talked about some sex differences earlier on. I'm wondering whether you've had a chance to look at that in the context of the pregnancies, because you know you've got female and male placentas.
This is ongoing.
Okay.
Yeah, of course. We are doing this in all projects right now. There is no sense anymore to evaluate together, but as it's, preliminary results was nicer to show together right now, but I agree that we might have differences, of course. And in the next workshop, maybe Victoria is going to be here with us to present.
Yes. Hi, Tanya. I was wondering if you're interested in following up the offspring as well, because you mentioned like you have higher resorption, and I was interested to know what is the litter size and if that might also affect growth after, if you
Flavia and Marcela is just nodding. Hear you. Yes, of course, we are interested, and it's also ongoing. It's very funny when you work with mice and you want to get them pregnant, because when you just leave them in the lab, they are always pregnant. But when you have to determine exactly when they need to get pregnant, and with the diet is a nightmare. So sometimes we put, I think the numbers were not there, how many they were there, I don't know, if how many we actually put on a diet and how many got pregnant after each experiment. When we planned the experiment, we had no idea how difficult it's to get pregnant, or the air condition is too cold for them, or some noise outside the facility, and they don't get pregnant. Now in the summer, usually it's even worse to make them, so it takes a while. People are nodding. Everybody agrees with me.
But do you have that information regarding litter size, and if it could be an issue, like you're seeing that fetal weight is affected, so they're it's lower, right? But then if you have like a very small litter size, couldn't it
I think
be
the
a
deficient
factor?
group has less, the litter size is smaller, and they are also smaller. So it's not the size of the litter, the number of fetus that are, it's not impacting the size. It's
No, I mean afterwards, if you follow up the offspring, if it could affect growth.
No, we kill them to get the placenta. So it has to be after Victoria. Yes. We plan to do in now all the models, but we never get there.
No.
She's interested.
Yeah, what I mean is next steps. If you're interested in following them up, wouldn't it be an issue? Is it that little? If because resorption is higher, does
we
it
are
become
interested.
like a?
I think we stop the pregnancy, of course, but I think that we will be able to follow up after. Yeah, I think so. Yes.
Tanya, this is the, I was going to ask about the one hydroxylase in the mouse placenta, because I know that's in humans. Of course, the enzyme is there, but what's the activity, and also how it kind of regulates the transfer of activated vitamin D to the fetus. So that's kind of amazing that the mouse reproduce actually the human what we see vitamin D metabolized.
We use these enzymes and genes right now to actually correlate the effect of the diet, because one of the struggles is actually to measure the vitamin D in the serum of mice. It's like virtually no sample, and so we are working in a collaboration with Marina. What's this last name? I don't remember.
Marina Amaral.
Thank you. So Marina Amaral, that we are already able to detect. It's ongoing. It's actually working, and we are going to have data soon to measure the vitamin D after the diet treatment.
So, Jorge, I know you have a question, but just how about the mother? Because the reason I'm asking is, so it seems to me that vitamin D is the cure for everything. Of course, COVID was, but for hair loss or for bad breath. But how about insulin sensitivity?
Everything, COVID.
No, but
you can use,
how
it's
about
a magic.
Yes, but
Hold on,
how
magic pill.
about insulin sensitivity and glucose metabolism in the mother? Because that might explain not only vitamin D deficiency, that decreased litter size or
But
weight.
to study the
Yes.
the pregnant female.
Yes.
We know we took we have the liver and the kidney of the females, but we didn't touch them yet. But we can do. That's what we have: liver and kidney. Yeah, it's we are not, we don't work with metabolic pathways very much much in the lab. It's so we need another collaborator to do the metabolic stuff. But yes, we have the liver.
Okay.
It's possible.
Okay.
Yeah.
Okay. Jorge? Yes.
Yeah.
Okay. Yes.
Yeah.
Yes.
It's ongoing. Yeah. Okay.
Hi, Tanya. I already know that vitamin D is one of the safest micronutrients to use in high doses, at least in human. But when you show the table with the resorption number, if I'm not wrong, the supplemented group had the most fetal deaths at the 10,000 IU's dosage. So I'm thinking, we are always talking about the deficiency. Do we have any kind of...
So now we have one, the first issue. Thank you for the question. So the first issue, you are in the nutrition lab, and I'm in the endocrinology lab, right? So vitamin D for me is a hormone. And if it's a hormone, it's not safe to supplement. So I have to disagree with this idea that I know that that's why I started nutrition lab, and because there is my truth, your truth, and the real truth that we don't know yet.
You know, I get it.
I probably the excess of vitamin D would be safe for some effects of vitamin D, but not even for bone health. The excess of vitamin D is safe, so be very careful. Here we have physicians prescribing lots of vitamin D, but usually they are based on the fact that we are actually insufficient or deficient, even with all the sun that we have here. But in some countries, like in the US, they buy vitamin D easily. For us here, it's not a problem right now. It's very expensive. If you actually look for vitamin D to take, it's an expensive hormone to use. But in the US, it's very cheap to buy. You buy big bottles of vitamin D. So I would say that it might be safe for some vitamin D effects, but not for every vitamin effects. Also, we are looking at the 1-hydroxylase and 24-hydroxylase in the placenta. That means that in all the tissues that we have these enzymes, the local regulation might be different. So we are going to take the 25-hydroxylase, and then if in one tissue it's going to be inactivated or activated, we can say exactly right now. We have a long way to actually understand vitamin D in every tissue. That's my opinion.
No, I agree with what you said. I just said that it's the safest because as a nutritionist, we are always seeing prescription of, I don't know, capsules with 50,000 IUIs. And in United States, we see even more high dosage because everyone, at least in these sites of the vitamin D, treat it as you don't have any kind of problem. You don't have any upper limit to the dosage. So when I saw this fetal deaths, I thought that maybe 10,000 could be too high, too much. I don't know.
Yeah, well, we don't know. also we try the 10 times higher than the normal weight because we had to start in in some way to supplement. I actually I don't know. I think that the data with miscarriage, reabsorption, and we need more. In a special, our control group, we need a stronger control group to make sure. So in the next workshop, Victoria is going to present, not that preliminary data.
Okay, thank you.
Thank you. Yes. Yes. It's good that we have all the time the workshop, so you can confirm. Okay. So.
Any more questions? No? Okay, thank you so much.
Thank you.
Yes.
So I'm going to stay here now to introduce the next speaker. So the next speaker is Dr. Enrico Bois from the Federal University of Minas Gerais. Dr. Enrico, first of all, he's my friend. So it's to say something personal. So, but he's great researcher in reproductive field. He has been working in the reproduction field more time than I have because I'm original thyroid person. And at the beginning of the project, everything that I learned about the placenta, it was from Enrico. And as a hobby, he likes very much food from Minas Gerais. That is a very good food, actually, and gardening as well. So, Enrico, please welcome.
Well, thank you very much I would like to thank Dr. Tatiana and Tanya for the invitation of being here. It's always a pleasure. It's very nice to see a full house, right? So today we're going to talk about infection disruption of placenta multi-drug resistance transporters and its implications to fetal drug exposure. Let me see if I can make this work. Yes. So we'll be talking about infection during pregnancy. Tanya. Yeah, infection during pregnancy. And we'll be discussing points on how the placenta functions as a barrier to drugs and teratogens. And we'll be discussing our findings on bacterial, viral, malaria, and polymicrobial infection modulating placenta multi-drug resistance transporters. So let's begin with infection during pregnancy. And when you think of infection during pregnancy, we have to think that it may be transmitted to the fetus through different mechanisms, but two most important ones are the transplacental transmission and through ascending infection by invading the cervix, right? And reach the fetus. And at the maternal level, it may impact EZs. Do you have? Okay, so at the maternal level, it may increase inflammatory response, it may induce sepsis, cause respiratory distress, and even death. Oh, okay, so, but it impacts the mother, but it also may impact the placenta because it may induce decreased nutrient and oxygen transport, which will lead to fetal growth restriction. Okay. Okay, so, and this will lead to fetal growth restriction, and it may also induce preterm labor. Preterm labor is a major global health issue, and fetuses born with preterm labor may develop birth defects such as blindness, deafness, microencephaly, neurodevelopmental disorders, and even fetal demise. So it's a little bit mechanic. So when you think of the infective agents that may impact pregnancy, we'll think of the torch infective agents. And the most important torch pathogens are the Toxoplasma gondii, which will produce toxoplasmosis. We have rubella virus, cytomegalovirus, herpes virus, but we have other pathogens as well that are the bacterial vaginosis, those that are involved in bacterial vaginosis. We have also the brucella causing brucellosis, dengue virus, Zika virus, chikungunya virus, HIV, the influenza virus, and also the Plasmodium falciparum that causes malaria in pregnancy, and even SARS-CoV-2. So from a global perspective, preterm birth occurs in approximately 15 million% of pregnancies every year. And this comprises approximately 10% of all pregnancies and accounts for approximately 50% of long-term neurological morbidity. Therefore, preterm labor is a major global health issue, and understanding the causes of preterm labor, as well understand the impact that preterm labor induced to the fetus is very important. So now let's talk about a little bit on how the infective agents are recognized by the body. So we have the innate immune system that has these toll-like receptors, the toll-like receptor 3, okay, toll-like receptor 3, toll-like receptor 7 and 8, and toll-like receptors 2 and 4. So, toll-like receptors 3 and 7 and 8 will recognize viral particles, whereas bacterial products and protozoa products will be recognized by the toll-like receptor 2 and 4. So in response to the recognition of those infective particles, the cells will respond producing a local release of specific cytokines, specific cytokines, and that will produce immune cell infiltration and also produce inflammation. So when you think of the maternal-fetal interface, we have to remember that they also express toll-like receptors, and we have a number of toll-like receptors being expressed at the placenta, at the decidual, the fetal membranes, and even the yolk sac. So in the placenta, we have a number of different cell types that express different toll-like receptors, and we also have those, as I said, in the decidual fetal membranes, and they are able to recognize infective particles, and they are also able to mount a specific inflammatory response to combat the different types of inflammation. Okay, so how the placenta barrier works, so how does it function as a barrier to drugs and teratogens? So, the functional units of the placenta is the chorionic villi, and the chorionic villi floats within the intervillar space of the placenta, which is enriched with maternal blood. And externally, the chorionic villi has the syncytial trophoblast cells, right, that is in direct contact with the maternal blood, and in the inner core of the chorionic villi, we have those fetal capillaries. So when you think of the placenta barrier to drugs and toxins, we have to think of the syncytial trophoblast and then also of the endothelium. But it's very important when I think of the protection that the placenta provides the fetus, we have to think of the maternal gestational age in which the placenta is providing protection. Why? Because the syncytial trophoblast is thicker in early stages of pregnancy when compared to later stages of pregnancy, as well as the fetal capillaries, they are much lower in early stages of pregnancy when compared to later stages of pregnancy. And this is important because it favors growth of the fetus towards the end of pregnancy. At the end of pregnancy, the fetus needs what? More nutrients, more glucose, more amino acids, and this favors fetal growth towards the end of pregnancy, but it may also favors what? Favors exchange of xenobiotics and environmental toxins towards the end of pregnancy because the placenta is more leaky. So here we can see that fetal mice exposed to tritiated digoxin, those fetal mice, they accumulate more digoxin at the end of pregnancy when compared to earlier states of pregnancy. And this effect was not observed in the maternal brain, demonstrating that this was a effect specific to the placenta. And this consists with the general embryo vulnerability idea of teratogens during early stages of pregnancy. So if the embryo is more susceptible to drugs and teratogens in the early pregnancy, in states of pregnancy, then it's because you have a thicker syncytial trophoblast layer, you have less fetal capillaries there in early stages of pregnancy, but we don't only have those morphological changes in the placenta. We also have the placenta enriched with different transporter systems. And that's when we talk about the ATP binding cassette transporter systems. So what happens is that this transport system, which we call ABC transporters, they will efflux their substrates from within the cytoplasm towards the extracellular space, right? So that's why they are called the multi-drug resistance transporters, or also MDR, because they are called multi-drug resistance transporters because in cancer cells, we have anti-cancer drugs that cross the cell membrane towards the cytoplasm, but in cancer cells, you have higher expression of ABC transporters that will pump those substrates from within the cell towards the extracellular space, decreasing their levels, therefore causing also drug resistance to those cells. Okay, so you may think, oh, in cancer cells, they cause cancer resistance, but are they important in biological barriers? Yes, they are very important because they may limit transfer of their substrates from one side to the barrier to another. And there are over 50 ABC transporters identified, subdivided in 7 different subsets, ranging from ABCA to ABCF, and the 2 best described ABC transporters are PGP and BCRP, P-glycoprotein encoded by ABCB1, and BCRP encoded by ABCG2. So we can see that there are a number of very important physiological and pharmacological substrates of obstetric relevance. I'm still fighting this. Yeah, because it's very strange to switch. Okay, so for the physiological substrates, we can see that we have different hormones, just like aldosterone, cortisol. We have different cytokines, different chemokines, and we also have nutrients such as folate, right? And for the pharmacological substrates, we have a range of antibiotics, antidepressants, antivirus, and anti-HIV drugs, synthetic glucocorticoids, and also a number of toxins, demonstrating that those transporters, they are very important for biological barriers, include the placenta. So when you think of the role of ABC transporters promoting fetal protection, we have to think where they are localized at the syncytial trophoblast, which is the major barrier at the placenta. So PGP and BCRP, they are expressed at the apical membrane of the syncytial trophoblast facing the maternal blood, which is enriched with toxins, xenobiotics, and also steroids. So as we can see, there is higher concentration of toxins of those substances in the maternal blood when compared to the fetal circulation, because PGP and BCRP, they are active transporters that will prevent the transfer of those toxins from the maternal blood to the fetal circulation, because they pump back all of those substrates, therefore providing fetal protection. So let's talk now about our findings on bacterial infection and placental MDR transporters. So when you think of infection during pregnancy, of bacterial infection during pregnancy, we have to think that pregnant women are at greater risk of ascending bacterial vaginosis, because during pregnancy there are a number of immunological adaptations that pregnant women has to go through, and that may make them more susceptible to develop bacterial vaginosis that may cross the cervix and reach the fetus. And infection during pregnancy may induce pregnancy loss. It may induce miscarriage and stillbirth, and most importantly, chorioamnionitis, which is the infection of the fetal membranes and also infection of the placenta, the umbilical cord, and or the fetus. And chorioamnionitis is highly related to preterm birth and neonatal sepsis. And then we asked ourselves, okay, so if chorioamnionitis disrupt the placenta, may cause preterm labor, is it direct, does it change placental multi-drug resistance transport or activity? So we use LPS as a model. LPS is a constitute of the bacterial cell membrane, and it binds to TLR4. And TNLI, Dr. Matthews' lab, where I did my postdoc, investigated if toll-like receptor 4 is expressed in first and third trimester placenta, and she identified that TLR4 is expressed in the first and third trimester placentas, and then using trophoblast explants of first and third trimester exposed to LPS for 4 or 24 hours, we observed that there is decreased expression of ABCB1, which encodes for PGP, and ABCG2, which encodes for BCRP. So there is decreased expression for ABCB1 and ABCG2, followed by decreased protein expression of PGP and BCRP after LPS exposure in first trimester placental explants, but not in the third trimester placental explants, demonstrated gestational age effects of LPS modulating placental PGP and BCRP expression. Okay, so next protein levels were downregulated. So what happens with PGP activity, for instance? So we decided to measure PGP activity, and then we inoculated LPS in black 6 mice for 4 and 24 hours at the gestational days 15.5 and 17.5. And then we used digoxin. Digoxin is a specific PGP substrate, and we used tritiated digoxin to evaluate PGP activity. And what we found was that there was increased fetal accumulation of digoxin 4 hours after LPS treatment, but not 24 hours after LPS treatment. This demonstrates, okay, so if you have greater accumulation of digoxin in the fetus, it demonstrates that PGP is actually inhibited. So its activity is inhibited 4 hours after treatment, but not at the gestational day 17.5. There were no difference in digoxin accumulation at both gestational ages. But we can see that there was increased interleukin 6 greater amounts of IL-6 at the maternal blood 4 hours after LPS exposure, but not at 24 hours, suggesting that a cytokine response to LPS acutely was, in fact, changing PGP activity in the placenta. However, at the expression levels, further studies that we did here, we did not find differences of LPS in changing both PGP and BCRP expression in the placenta, demonstrating that this effect was more related to PGP activity. So as to viral infection, viral infection may also induce preterm labor in the mother. It may induce different kinds of disease in the mother. It may affect the placenta, and it may also affect the fetus and cause preterm labor. And there are different viruses. Well, as you think, well, okay, we have more than 200 viruses, if I'm not wrong, described to impact the human. But if you think, we have 6 to 8 or 9 viruses that actually can reach the fetus. So the placenta does actually an amazing job preventing fetal infection. But the most important viruses that are related to poor pregnancy outcome are cytomegalovirus, influenza virus, herpes simplex virus, Zika virus, rubella, and varicella zoster viruses, and also SARS-CoV-2. As we've seen before, SARS-CoV-2 may impact pregnancy, it may reach the fetus, so we decide in the beginning of the SARS-CoV-2 pandemic, we investigated whether placenta tissue would express ACE2, which is the major cell entry protein for SARS-CoV-2. And then we collected preterm labor placentas with chorioamnionitis, and then we identified that ACE2 is expressed in the syncytial trophoblast cells. It is expressed at the decidua, at the extravillar space of the placenta, and as well as in neutrophils and macrophages that are invading the placenta. So we thought by that time that actually neutrophils and macrophages invading the placenta could be delivering SARS-CoV-2 to the fetus. But as I mentioned before, there are very rare occasions, and we don't know why, the fetus may be actually infected by SARS-CoV-2. We also were one of the first groups to demonstrate that ACE2 and TMPRSS2 are expressed by the placenta at different gestational ages. And more recently, we demonstrate that brain endothelial cells, which are derived from the developing brain at early and late gestation, and mid-gestation, also express ACE2, TMPRSS2, and furin, demonstrating that the fetus, since early gestation, may have neuroinvasion. Okay, but just to show that SARS-CoV-2 may also impact pregnancy. So, and then we asked ourselves if viral infection could change drug exposure through alteration in PGP and BCRP. And there are many toll-like receptors that can recognize viral infection. Among them, we have toll-like receptor 2, 3, 4, 7, 8, and 9. However, toll-like receptor 3 and toll-like receptor 7 and 8 are the best studied, toll-like receptor study for recognized viral particles. TLR3 recognized double-strand RNA viruses, whereas ssRNA, whereas TLR7 and 8 will recognize single-strand RNA viruses. And there are two viral mimics that we use a lot to mimic viral infection, which is poly-IC, that binds to TLR3, and it is TLR7 and 8 that binds to, is recognized by ssRNA. However, the effect of ssRNA viruses modulated placental MDR expression and function is yet to be determined, but we have done some work in studying poly-IC as a model to as a model of viral infection. So we collected first and third trimester placenta. We did placental explants, and we treated them with poly-IC, and investigating that first and third trimester placenta also expressed TLR3. However, we did not see an effect of poly-IC modulating PGP and BCRP, both at the messenger RNA and at the protein level. So this demonstrates that viral infections are less likely to impact viral exposed to drugs and toxins. So then we wanted to verify if the same would happen at the activity level. So what did you do? We used poly-IC. We treated pregnant mice with poly-IC after 4 and 24 hours. And then after 4 or 24 hours, then we use digoxin as a model, as a specific PGP substrate. And then we saw that in these mice, there was increased levels of IL-6 for hours after treatment, but 24 hours after treatment, we did not see a cytokine response. But we did not see any effects on placental PGP after poly-IC treatment, demonstrating that, at least in the mouse model, poly-IC does not change placental PGP activity. But what was interesting is that we also investigated the digoxin levels in the fetal brain. We have PGP and BCRP being expressed at brain endothelial cells of the blood-brain barrier because they are very important to provide protection of that tissue. And for our surprise, there was increased levels of digoxin in the fetal brain 4 hours after poly-IC treatment, but not 24 hours after poly-IC treatment, demonstrating that even though poly-IC did not change placental PGP activity, it did change activity of the blood-brain barrier in the fetal brain. Therefore, alterations in brain permeability to PGP substrates may be one of the mechanisms that viral infection may cause neurodevelopmental disorders. So Shirley Andrade investigated whether Zika virus infection could also change PGP and BCRP activity expression at the placenta. And for that, she used immune-competent mice and immune-incompetent mice, and she infected those mice with high or low doses of Zika V and at gestation day 12.5, and sacrificed them on gestation day 18.5. And what she observed was that Zika virus in high or low doses and in immune or immune-competent mice would decrease PGP and BCRP expression at the labyrinth zone, which is the major exchange site in the mouse placenta, demonstrating that Zika virus has the potential to increase fetal and toxin drug exposure during pregnancy. So how about malaria infection? So malaria is a major health issue because malaria in pregnancy can induce maternal anemia, stillbirth. It may also induce spontaneous abortion, preterm labor, and induce low fetal and placental weight. And it is estimated that 11.6 million pregnant women in the World Health Organization Africa region is exposed to malaria in pregnancy, has malaria in pregnancy. So it has a major impact on pregnancy outcome in different countries of the world. So it causes preterm labor. It is an infection. It is modulated TLR. We saw evidence that both PGP and BCRP may be modulated by cytokine storm. So then we developed a model of mouse of malaria-induced preterm labor, where we developed a model of malaria infection in the mouse during pregnancy that would induce preterm birth. So what we observed that malaria infection during pregnancy can mount a huge pro-inflammatory response and also decrease PGP and BCRP expression in the mouse labyrinth zone, both at the protein and at the messenger RNA level, suggesting that malaria pregnancy has the potential to increase fetal drug exposure to drugs and toxins. So finally, we wanted to investigate whether polymicrobial infection would also alter placental MDR. So what is important here is because ascending polymicrobial infection is one of the main causes of preterm labor, which generally follows a previous ascending vaginal infection. So during pregnancy, there's higher chance of vaginal infection. This vaginal infection may ascend and infect and induce chorioamnionitis, and it is usually not induced by one single kind of bacteria, but by a polymicrobial infection. So Guinevere Império, she collect placenta, preterm delivery placenta with and without chorioamnionitis, and she investigated the expression profile of 50 ABC transporters. And she identified that there were 5 ABC transporters upregulated in preterm delivery with chorioamnionitis placenta compared with those preterm labor without chorioamnionitis. Here it's important that we wanted to investigate and compare preterm placentas with and without infection, but we did not compare them with term placenta. Why? Because there is a gestational dependent profile of expression of those transporters. Okay, but then we collected, we did some Western blots, but we did not find differences at protein levels for PGP and BCRP comparing those two groups. But we have both PGP and BCRP being expressed in other cell types of the placenta, and what we are really interested were the levels of PGP and BCRP at the syncytial trophoblast, where they provide their fetal protection action. So we did immunohistochemistry for PGP and BCRP, and there was an increased immunostaining of BCRP at the syncytial trophoblast, and there was, in accordance with ABCD2 levels being increased. However, we identified that there were lower levels of PGP at the syncytial trophoblast in preterm delivery of chorioamnionitis placenta compared with those without chorioamnionitis. But I don't know if you remember, we identified increased messenger RNA levels of ABCB1, but now we identify decreased PGP protein levels, not decreased, sorry, increased PGP, no, sorry, decreased PGP protein levels in the syncytial trophoblast. So there was a mismatch between messenger RNA protein levels. So then we identified 4 microRNAs that are known to decrease PGP translation, and we observed that there was increased expression of MIR3315P. And we also did a correlation with the chorioamnionitis degree of our cohort, and we observed an increased correlation of MIR3315P, demonstrating that as the degree of chorioamnionitis increased in our samples, there was increased expression as well of MIR3315P. So although we observed increased ABCB1 messenger RNA level, protein levels were decreased, probably due to higher expression of MIR3315P. So what we conclude that polymicrobial infection can induce preterm labor with chorioamnionitis, and that alter placenta PGP and BCRP expression could also alter biodistribution of drugs and toxins in preterm delivery fetuses. So we conclude that PGP and BCRP are key components of the syncytial trophoblast barrier. We conclude that different infective agents modify placental MDR in a gestational age, trophoblast type, and species-specific dependent manner through activation of specific TLRs in the placenta. We also conclude that gestational Zika virus and malaria pregnancy mouse models of infection can impair MDR in the placenta, whereas in humans, placenta and MDR response are variable, most likely resulting from the polymicrobial nature of the human treating infection. We also conclude that alterations in placenta caused by infection may adversely affect pregnancy outcomes and impact maternal and neonatal health. So if you're interested in what's been described here, these are the results that are reviewed in this review paper written by Shirley Andrade. And I'd like to thank all of our collaborators, the past students and the present students, all the technical staff. I would like to thank also Dr. Matthews, Tanya Ortiga, Flavia Blois, and Fernando Reis for their collaboration and friendship all over the years, and also all of the funding agencies. And questions, and if I could ever help anyone, please contact me. Thank you.
Thank you, Enrico, and it's open for questions.
Hello, great presentation. I just wanted to know, especially in your animal models, have you been able to separate it by sex and see if the response to, for example, the Zika virus is the same in males or females?
Well, we did not in the beginning, but as Tanya said, this is ongoing.
Ongoing, okay.
But I mean,
in the Zika virus, we already have the preprint out with the.
oh yeah, it's true.
So
But yeah, and yeah, it's very important. We did not find we found major differences, but it was more related to metabolism. I don't really recall the specific changes, but we were discussing the other day, and that there are, when you talk about infection, the male and the female conceptors respond very differently. Usually male conceptors, they have a greater response to inflammation and infection, which can be more detrimental. So, but that is not always the case, and it depends on what kind of insult they are being exposed to.
Thank you.
More questions? So let me comment. So the malaria and LPS model, Klaus is doing with the females and males separated, but he couldn't be here. I want him to present the data today, but he couldn't. And the Zika virus is Daniela's paper that it's preprint right now.
Enrico, great talk. Thanks for the lecture. No,
I'm
you're
sorry,
very.
I got a little bit, this really stressed
No,
me.
that's.
But
You don't seem
I know,
stressed.
I know, but I was. So. But it's not it's anybody's thought,
No.
right?
So this just a curiosity, and maybe you have tried. So bioactive compounds, polyphenols in general, and I saw that flavonoids.
Yeah.
So how do you see it? They are xenobiotics, right?
Yeah, they
and
are.
And how they, because the liver treats them as, secondary metabolites and so. And how about, the effects on MDR and
yeah, what happens
infection.
is people don't really investigate the role of PGP of ABC transporters for their physiological substrates, but they are expressed in the liver, they are expressed in the kidney, they are expressed in many tissues. So, and we don't know, I don't know exactly how they behave in the placenta and metabolizing, right? But, PGP and CRP, they are present also at the blood-brain barrier. When you are, for instance, during infection, if you are decreasing their activity, their expression, you may also have decreased transport, decreased accumulation of, for instance, folate, micronutrients. So this is also very detrimental to the fetus. And it may also contribute to, as I said before, to the neurodevelopmental alterations that we see, right? So the next step of our research, at least something that I want to do, is in those models, is try to mitigate, but on what to mitigate in a way that you can do, showing, of specific compounds or environmental changes. We are still ongoing investigating, right? But for sure, for me, the physiological alteration, because PGP and BCRP exist there for not millions, but a lot of time. And those proteins evolved to protect, to control transfer in biological barriers that transferred our physiological substrates. And of course, environmental toxins. But we studied more focusing on drug exposure, and drugs exist, 200, 300 years? So this is a very important question.
The reason I asked is because of the many flavonoids and not polyphenols are used in cancer cells,
Yeah.
and to decrease proliferation and induce apoptosis, and some of them are known for a fact that they act on PGP, and so
For sure.
Yes, that's and there are many common pathways in cancer cells and trophoblast cells. So,
But you know what? It's interesting. It's because in cancer cells, we always know, but in many cases, increased expression of PGP. But usually, you increase expression of PGP when the cells are proliferating a lot, right? So it's not to. Yeah, and then it goes down. I mean, stops proliferating, and you see decreased PGP levels towards the end of pregnancy. I did not show here because of the time, but PGP is more expressed in early stages of pregnancy than later stages of pregnancy. There's less proliferation, there's more differentiation. Yeah. Thank you. So I'm waiting for you to invite us to study PGP and BCRP in your models of endocannabinoid mice, rats.
So there is a relationship,
For
and my
5
question is
years
about
now,
it.
I'm waiting and nothing.
So I was wondering if you thought about measuring inflammatory molecules derived from lipids in these different infection models, such as prostaglandin and things like that, or if the literature shows that there is an alteration in this lipid pro-inflammatory pathways in these infections related to preterm birth.
We did with Mila, and there was found that the LPS would change some of the some lipid accumulation transfer in the placenta. I don't remember which, but I remember I was astonished on how inflammation would alter the whole lipid, metabolism and transfer in the placenta. And with respect to those lipid pro-inflammatory metabolites, we did not, but this is something that we could absolutely do, and I'm sure we find differences.
The reason why I'm asking is because the endocannabinoids, they can be metabolized in arachidonic acid, which will derive these lipid inflammatory pathways, and they can also be metabolized directly into these pro-inflammatory molecules. And it seems there is a time course increase of endocannabinoid levels in placenta across physiological gestational age. And it increases with the gestational age, and it seems to be related to preterm birth. Maybe.
So they're thinking choriamnitis placenta, you would have an increase of the system?
Yeah.
But you said that it is anti-inflammatory, but in the placenta they are pro? Oh, they're a pro.
the products derived from the metabolic pathway because of the arachidonic acid. So the lipids can have these pro-inflammatory producing prostaglandin, for
Okay,
example,
yeah.
as a source to produce prostaglandin. And the CB2 action is another story. It's
No,
an
I just
anti-inflammatory. You were
remember,
right.
prostaglandin has been shown to increase PGP activity in the placenta. So you are in the right way.
Thank you.
Thank
Congratulations.
So thank you, Enrico. And now let's go to our last speaker, but not least. So Dr. Steve Matthews is a professor at the University of Toronto. He has an amazing number of publications. He has worked with placenta and glucocorticoids and also fetal brain. And he is amazing in make collaborations all over the world. So he travels a lot. And to say something personal, that is not exactly what he told me to say, but I'm going to compliment that he loves Rio. And besides that, he also likes canoeing and also sailing. Let's see if he can master the pointer.
Oh, this is the biggest challenge.
Yeah, I'm just going to disclose that I actually have this pointer and I use and I teach with it. That's why I could master. And all my students that always ask me for the pointer, I always look at them and say, you know, my pointer is not a good idea to just try to use it. And now they understand why I never want to Steve is going to master it.
There we go.
See?
Right,
Thank you, Steve.
Thanks very much, Tanya. And thank you, Tanya and Tatiana, for organizing this great symposia today. It's wonderful to see so many people here in the audience. I know you've had a long day. I'm going to keep things simple, and I'm going to try not to present too many data slides. And I want to tell you a bit of a story, actually, as I go through the presentation today. So, first of all, I want to acknowledge the folk who have really helped me on the journey that I've had over the last 30 years of doing research. I think you recognize this guy on the top here, Enrico. Enrico and I have worked together now for about 11 or 12 years. And together with Tanya and Fernando, we've undertaken a very successful collaboration. And today I'm going to talk not really about transporters. I'm going to come right at the end to talk about transporters, but I'm going to talk about the work that we've been doing on programming. So I'll first start by introducing the concept of early life programming, and we've already heard earlier today about the area of DOHAD, or developmental origins of health and disease. I'm going to talk about it much more in the context of glucocorticoids and the developing fetal brain. I'm going to talk specifically about behaviors and HPA function and how these early exposures impact that. I'll talk about how it is that glucocorticoids can lead to long-term changes in function of the brain, function of the HPA axis, and functional behaviors, because they don't just have effects in one generation. We've now shown that effects can pass across 3 generations, and not only are they transmitted down the maternal line, they're transmitted down the paternal line. So there's a lot of really interesting work going on in the area of transgenerational transmission, and glucocorticoids are being used as a model here. I won't have much time to talk about that, but I can certainly answer questions that you have. And then at the end, I'm going to talk a little bit about brain vulnerability, and I'll talk a little bit about the drug transporters and some of the very recent work we've done in that area. So I don't think I have to convince this group that environments during early life have long lifelong impacts on trajectories towards health or disease, learning capability, and social function. And there's been a huge interest in this from the public, and a lot of articles in the sort of mainstream media identifying how early environments can lead to very long-term impacts on our health throughout our lives. And there's a huge literature. When I started in this field, that you could probably, there weren't too many publications in the area. But now, if you look, there's thousands of publications that essentially are linking early exposures to long-term outcomes. So, and a lot of focus has been placed on the mother and the environment of the mother. And this is a challenge because, in fact, we know now that the environment of the father is important. As important, we don't know, but we're starting to identify a lot of the mechanisms that link paternal adversity or paternal environments to offspring health that are actually independent of the mother. So there's a lot to be said about the paternal environment that's important. But what we know is that anxiety, stress, hormones, drugs, placental insufficiency, undernutrition or overnutrition, obesity, or altered postnatal care can lead to long-term changes in cardiometabolic function, including hypertension, insulin resistance, and diabetes, and also obesity. Some cancers, particularly cancers like breast cancers, have been linked to early environments. But then an area that I'm more interested in my research is around brain development, learning ability, attention deficit hyperactivity disorders, depression, and self-regulation, how an individual interacts with their environment. But so we've been interested in these associations, but in fact, what we're more interested in is what is it that links the early environment to long-term health outcomes? What are the mechanisms? How long do the effects last? And if we can understand these things, can we develop interventions that either prevent or reverse these effects? And so we've spent a lot of time thinking about this over the last several years. I want to then just flip over and talk about one of the models that we've used to ask questions around the early environment. And we've been particularly interested in glucocorticoids. And if we're to understand the role of glucocorticoids during development, we need to understand a little bit about the regulation or the development of the pituitary adrenal system in the fetus. And we know that in late gestation, the fetal HPA axis, so here we've got the paraventricular nucleus, which produces CRH and AVP, which then drive the anterior pituitary corticotroph to produce POMC, which is then processed to ACTH. ACTH then is released and drives the adrenal cortex to produce cortisol. This is the classic textbook stress response, right? You're all familiar with this. We know that this system is functional in the fetus. We know that the fetus can respond to stress by activating this system. We know that there are very important feedback mechanisms that feedback to suppress activity so that they create a homeostatic process and feedback regulation. And they do so at two primary receptors. They do so at the level of the glucocorticoid receptor, which is red, and the mineralocorticoid receptor, which is green. And this structure at the top that I haven't told you about is the hippocampus. And the hippocampus is present, is a limbic structure in the brain. It contains very high levels of glucocorticoid receptors and mineralocorticoid receptors, is incredibly important in regulating behaviors, learning, memory, all sorts of higher function. But it's also very important in regulating the pituitary adrenal system. So we know that this whole system is functional in fetus. We know that it can be activated. But what we also know is that in late gestation, there is a very dramatic increase in pituitary, hypothalamic pituitary adrenal activity in the fetus, and also transfer of maternal cortisol to the fetus, which leads to a massive increase in cortisol in nearly all mammalian species at the end of gestation. Now, I'll ask you, what is this glucocorticoid surge doing? Anyone in the audience? And Fernando's not allowed to answer this because he's an obstetrician. What's that surge in glucocorticoid doing? That surge in cortisol? Come on. Come on. Lung development. Okay. So the surge in cortisol is really important in transitioning the fetus to becoming a neonate. The fetus has all of its organ systems developed, but they're functioning in a fetal way. It doesn't need to use its kidneys because the mother does, the placenta does all of that. It doesn't need to breathe, obviously, because the placenta does all of that for it. But what happens during this phase, this surge of glucocorticoid, is that you get final maturation of organ systems in the fetus. Now, if a baby is born before this surge, there can be lots of problems, because if a baby's born before the surge occurs, you have a lot of problems with lung function, a lot of problems with breathing, high levels of mortality and morbidity. We heard about that earlier in the day around preterm birth. Preterm birth is very bad because your lungs aren't working properly if you haven't been exposed to this glucocorticoid surge. But so we've known for a long time and have focused very much on glucocorticoids being important in lung development, but actually I would suggest that glucocorticoids are equally important in maturing the brain, in transitioning the fetal brain to a neonatal brain. And this shows the expression of glucocorticoid receptors in this is a guinea pig brain. And the guinea pig, we use the guinea pig as a model for neurodevelopment because, in fact, from a neurodevelopmental perspective, it's about as close to the human as you can get outside using a primate in terms of the pattern of neurodevelopment and its relationship to birth. So if you're interested in neurodevelopment in the human, the guinea pig is quite a good model. This shows the expression of glucocorticoids in the hippocampus, the structure that I just showed you on that schematic, the dentate gyrus, the paraventricular nucleus, and this is the amygdala, which is important in other higher functions like fear and so on. And so you can see this is in late gestation, around 75% of the way through gestation. So this is when that normal glucocorticoid surge is about to happen. So you can see that the brain is primed. It's ready to see that surge of glucocorticoid. It's ready to go. So the glucocorticoid surge then, all of the machinery is in place to transduce that signal. And this is the point, that the brain is essentially hit by that glucocorticoid surge and it transitions. I don't have time to show you all of the data. If you're interested, there's a Nature Reviews that we wrote a number of years ago together with Vasilis Mercedes. But the bottom line is glucocorticoids are not only a master switch in the lung, they're a master switch in the fetal brain. And they do this switching through altering DNA methylation. So they result in what happens in the hippocampus is that with that surge, there's a massive demethylation and an increase in gene transcription such that over 1,000 genes upregulate following the glucocorticoid surge. So it's hugely active, very similar in many ways to what's happening in the lungs. The lung outcome is important because the baby needs to breathe. But obviously the brain is doing a lot of things as well. And it's not as clear when a baby's just born what the brain going on in the brain. The lung, it's easier to tell. But the bottom line is that surge is really important in all organ systems. And the brain is obviously particularly important and a focus of our research interest. So we know that the brain is impacted by glucocorticoids through that surge. But what happens if the fetal brain is exposed to a surge before it should be exposed to that late gestation surge. And how can glucocorticoids increase in the fetus? Well, we know that maternal stress and anxiety can increase cortisol from the mother, leading to an increase in fetal glucocorticoid concentrations. We know that fetal stress, I told you that the HPA system is all in place. Well, if you stress, physiological stress like hypoglycemia, hypoxia, infection, you can increase fetal cortisol concentrations. But you can also increase glucocorticoid by giving the mother synthetic glucocorticoids. Because glucocorticoids are so important in switching the lung, glucocorticoids are given, synthetic glucocorticoids are given to mothers who are at risk of preterm birth. And what these synthetic glucocorticoids are doing is in fact mimicking that glucocorticoid surge, so that when the baby is born preterm, it is able to breathe much better without mechanical ventilation if it's been exposed to glucocorticoid, because the lungs have been able to develop. But the question is, what's the impact of that glucocorticoid treatment in the developing brain? And I'll come on to that in a moment. We've looked at this question in many species, including the mouse, the guinea pig, the sheep, and obviously probably the most relevant model of, that we're interested in, the human, over many years. We know from the literature and from work that's come out from our lab and many other labs that there are long-term effects of prenatal glucocorticoid on the developing brain that can have lifelong or long-term outcomes. For example, we know that a premature exposure to glucocorticoids impacts brain structure, impacts behaviors, can impact neurosensory function, and it does so by influencing myelination of some of the major optic nerves, or the optic nerve and the auditory nerves, can have long-term effects on neuroendocrine function and cardiometabolic function. I'm just going to show you a couple of examples of that. So this is a study that was undertaken by Clemens Kirschbaum's group in Dresden in Germany. It was published about 10 years ago now, but it was a very important study. What they did in this study is they looked at children who were 6 to 10 years of age, who had been born at term, but who had been exposed to glucocorticoids in utero. So these were children that were born to mothers who had been identified as being at risk of giving birth prematurely. They were given synthetic glucocorticoids, but they then went on to give birth at normal term. So prematurity is not part of the equation here. All of the children on this graph were born at normal term. But there are 3 groups. There's a group here, the squares, which are the controls. So these were children that were born to mothers who were had no indication of preterm birth and went through a normal pregnancy. There were a group of women who were hospitalized during pregnancy, but didn't get glucocorticoids. And there are another, the red line here are children whose mothers had been given glucocorticoid, but were delivered at normal term. So these children were exposed to a Trier stress test. The Trier stress test is one of the most robust ways of activating the HPA axis in humans. And so in the case of the child Trier stress test, the child is asked to speak to 3 investigators, 3 researchers who are sat in a white coat, and they're asked to tell a story, which if you're 6 to 10 years old is actually pretty stressful. The adult version is far worse. You're asked to count down from 5001 in 7s. And that is tough. And that also increases your pituitary adrenal function. But it's very robust. So the bottom line here from this graph is that the children who had been exposed to a single course of glucocorticoid but had been born at term had an increased stress response. So they had a hyperactive pituitary adrenal function when they were 6 to 10 years old in response to this stress test. And interestingly, girls were affected more than boys. Both sexes were affected, but girls seem to be more affected than boys. So that was a really important landmark paper. We were able, with Vasilis Mercedes, who was a fantastic PhD student in our lab, what Vasilis did is he was able to model what we knew was happening in the human. We were able to model it in the guinea pig. And what Vasilis did was actually a heroic task. He looked across 3 generations. I'm just going to show you some data from the first generation. And of course, the advantage of using an animal model, as you all know, is that then we can ask mechanistic questions that you just can't ask using the human. So what Vasilis did is he gave maternal glucocorticoid. And in fact, in this study, he gave repeat courses, but we've shown similar outcomes with single courses of glucocorticoid. So this mimics what is being used in the clinic. And he gave the glucocorticoids to the mother. They then delivered normally at term, and he looked at the juvenile offspring. And we looked at endocrine function and behavior in these juvenile offspring. And what he saw was very interesting, was that when we exposed these animals to stress, we saw a very similar profile to what had been shown in the human, that the animals whose mothers had received synthetic glucocorticoid responded significantly greater to a stress test. And the stress test we use in the guinea pig is we put it into a novel open field environment with a light on top of it, and they run around fast and get quite stressed by that experience. Because it's unfamiliar to them. And what we saw is that there was a hyperactivation of the HPA axis in offspring whose mothers had received glucocorticoid. We also saw that the effects occurred in both males and females, and that this was associated not only with a higher neuroendocrine response, but these animals were much more active in the open field environment. So suggesting that they were hyperactivated in terms of their stress response. Interestingly, exactly the same was seen in regards to sex. So the female offspring were affected significantly more than male offspring. So it seems to be that the females are affected more in terms of neuroendocrine outcomes. So to sort of summarize a lot of work that's been done in my lab and other labs, we know now that the HPA axis, that early exposure to of the HPA axis to glucocorticoids can lead to lifelong changes in HPA function. This is important. Firstly, it's sex dependent. We've shown that now repeatedly in multiple studies. But it's important because if you reset or alter function of the pituitary adrenal system, you're going to alter a lot of systems, a lot of homeostatic systems, because the HPA axis is really important in the context of regulating metabolism, growth, repair, reproduction. So everything associated with kind of resource allocation, the HPA axis is important for, not just the stress response. So if you've changed its function by an in utero exposure, you're going to have a lot of long-term, lifelong effects. So we know that there are then impacts on behavior and learning. And we also know that if you alter HPA function, if you hyperactivate HPA function, you increase susceptibility for chronic diseases like insulin resistance, type 2 diabetes. So again, using an animal model, we can get much more mechanistic. So what are the mechanisms by which these glucocorticoid exposures lead to very long-term outcomes? Well, we know that glucocorticoids actually impact brain structure. They impact the way that neurons are born, so they impact process of neurogenesis, synaptogenesis, and we know that exposure to high levels of glucocorticoid lead to structural changes in the brain. But we also know that, as I said earlier, glucocorticoids are highly epigenetically active, and we became very interested in the potential mechanisms, the epigenetic mechanisms by which glucocorticoids can lead to these long-term changes in function. And we're particularly interested in DNA methylation, we did some work on histone modifications, which are interesting, but now actually our focus has moved more onto microRNA, but I'll show you a little bit of data. So this was a really simple study that was done in collaboration with Moishe Ziff in Montreal with another great PhD student, Ari Crudo. And what Ari did is we gave glucocorticoids at 2 time points, at day 40 and day 50. We then looked in the hippocampus, that structure that I introduced to you earlier. We looked in the hippocampus for changes in gene expression and DNA methylation that were associated with that synthetic glucocorticoid treatment. And this is before the natural glucocorticoid surge. Okay, so these are very glucocorticoid naive animals that we give that hit of glucocorticoid to, which is very analogous to what would be done in a human baby who was at risk of being delivered preterm. So we then looked at gene expression and DNA methylation. And when we looked at gene expression, we saw that a synthetic glucocorticoid hit on day 40 and day 50, when we looked in the hippocampus on day 52, 48 hours later, led to changes in gene expression in over 1,100 genes. Largely, these were upregulated as a result of the glucocorticoid surge, suggesting that they're having that kind of triggering effect that I alluded to earlier. When you give that synthetic glucocorticoid, you're simulating the natural glucocorticoid surge. So you're causing a triggering of changes in gene transcription. And when we look at DNA methylation, each of these columns here represents the hippocampus taken from a fetus from independent mothers. So this represents fetuses from 4 mothers. Guinea pigs only give birth to 2 to 3 offspring in our laboratory. So you can see here a single hippocampus, and going across each of the lines here represents a promoter, a gene promoter. So here we're looking at essentially DNA methylation in different promoters, and these have been bioinformatically aligned. And what you can see is that, and I should say that green represents lower methylation, red represents higher methylation or increased methylation. And you can see that with that synthetic glucocorticoid hit, in the large majority of cases, there is a decrease in DNA methylation associated with glucocorticoid. There are some genes where there's an increase in methylation in some of the promoters, but there's predominantly a demethylation. And this is consistent with the upregulation in gene expression that we see in the hippocampus. So the synthetic glucocorticoids are demethylating, upregulating gene transcription. Okay, so this is very similar to the natural trigger that we see at term. The important thing to know, and I'm not going to show you the data, is that of those 1,100 genes that are altered in transcription, and all of these altered DNA methylation, there's virtually no overlap with the genes that are altered if that surge occurs near a term. So the surge of glucocorticoids is activating a completely different set of genes because the hippocampus is a different stage of development. So it's not really surprising that long term there might be an impact on hippocampal function. So we've then spent a lot of time thinking about how glucocorticoids might impact the brain epigenome, and there's actually lots of routes, lots of mechanisms by which glucocorticoids can impact the developing hippocampus or the developing brain. We know, for example, and Tanya will be pleased that I mentioned the thyroid gland, that glucocorticoids actually, as I've said, they develop organ systems in the fetal brain or in the fetus, and one of the systems, one of the organs that they're very active in maturing in late gestation is the thyroid. We know that the thyroid produces thyroxine and T3, and that this is actually very epigenetically active. So glucocorticoids might indirectly activate or alter the epigenome through indirect mechanisms. But we also know that glucocorticoids alter placental transport of methyl donors to the fetus. They act directly at the GRE to change methylation. That's been shown in many studies. They're also able to alter the expression of many epigenetic regulator genes like DNMT and MBD2. They can alter the methionine cycle through changing expression of DNMT, and they alter levels of microRNA. So there's lots of ways in which glucocorticoids can actually modulate changes in gene expression that I've just shown you. And the fact that the HPA axis has been programmed means that this actually becomes a continuing conversation, because if you alter HPA function, and I've just shown you that glucocorticoids are so active epigenetically, that's going to have a knock-on effect that's going to sort of self-perpetuate throughout an individual's life. And that's something that we're very interested in, but it's probably very important in the long-term impact of prenatal glucocorticoids. So I've told you we started in the human, we went to the animal. I just want to go back quickly to the human, because the question is, do we see similar DNA methylation effects in the human? And one of the things that we have access to, one of our great opportunities we have in Toronto is the Ontario Birth Study, which I co-direct. And the Ontario Birth Study is embedded within our health system. We've got actually close to 4,000 pregnancies now, around nearly 14,000 biospecimens, and we've followed actually close to 2,000 children now up to 5 years of age. So we recruit women in early gestation. We follow them through their healthcare journey, through pregnancy, through to delivery. We then follow the children to 5 years of age, and we can start to link experiences that those pregnancies or that those women experience during pregnancy with longer term outcomes. And this is studies that were undertaken with Bona Kim, a PhD student in the lab. And one of the major advantages we have in Ontario is that we have blood spot screening at 24 hours of age. So all babies in Ontario have a heel prick and a blood spot taken 24 hours after they're born. Because our project because the Ontario Birth Study is embedded into the health system, we're able to add an extra card for research. And we developed techniques that allow us to actually use that blood spot to do an entire genome DNA methylation screen. So we can then extract the DNA and undertake DNA methylation. So Bona selected newborn, the blood cards from newborns whose mothers had been treated with synthetic glucocorticoids but had gone on to deliver at term. And there are actually a lot of women who get treated with glucocorticoids for being at risk of preterm birth that go on to normal term. About 40% of all women treated will go on to deliver at normal term. So it's not, there's quite a few children who go to term. So we're able to get their blood spots, and we then can do a case control, so we can match those pregnancies to other control pregnancies that we have within the Ontario Birth Study, and we match them on maternal age, BMI, parity, and fetal sex. And so then what Bona was able to do was she essentially extracted these blood spots. Here, she takes the blood spots, extracts them. She then does a bisulfite conversion, runs RBS and then sequencing. And she's then we're able to see DNA methylation across the entire genome. And this shows here, each of the columns here represents the different chromosomes. So you go across to chromosome 22 with the X chromosome at the end. And you can see that there was altered methylation in many, many genes. In fact, there were over 505 differentially methylated cytosines in the blood of these newborns that had received synthetic glucocorticoids when they were compared to control. That 231 sites were hypermethylated, 274 sites were hypomethylated. So there's a very profound impact of synthetic glucocorticoid exposure, and this was published in Translational Psychiatry last year. But the big question for us next was, so we've shown that in an animal model that we see this DNA methylation signature. We show now in the human that there's a DNA methylation signature. We can actually then flip back to the animal model and say, so we can see a signature in the blood of animals, we can see a signature in the brains of animals. How do they link to each other? And then how do those changes in the animal where you've got changes in blood and brain then map onto changes in DNA methylation that we've seen in the human? And this was a mammoth task of bioinformatics for Bona. She was able to, she looked in the prefrontal cortex, which is a very important area of the brain for behaviors that we're interested in. She looked at newborn blood from guinea pigs, and then she compared it to the DNA methylation screen from the newborn blood in humans. And she used an enormous array of bioinformatic tools to do the alignments, etc. But to cut a lot of work, several years of work down to 2 minutes, what Bona was able to show was that there were actually some really unique signatures. If you just compared DNA methylation across the different sites, there was nothing that really stuck out across all of the brain and the newborn blood. But if she focused then on a few transcription factors, a few transcription factor binding sites, she identified that there were 2 transcription factor binding sites, PLAG-L1 and TFAP2C. And these transcription factor binding sites were differentially methylated in the brain, in the blood of the human, and in the blood of the guinea pig. So there's commonality. And so we can actually predict prenatal glucocorticoid exposure based on looking at the methylation at these 2 transcription factor binding sites. And this is very, very new data that we haven't published. And what Bona's interested in doing is sort of creating some learning tools so that we can actually predict prenatal exposures to adversity, not only glucocorticoids. So this was a very recent and early, finding. So the idea here is that we've perhaps identified an early blood biomarker of prenatal glucocorticoid exposure. If we can do that, then we can also identify newborns that have been exposed potentially to high levels of adversity in utero. And we know that those newborn are going to be at risk of developing neurodevelopmental outcomes. So we can maybe start interventions early. And so we can really do some predictive analysis. And we're just starting to look in the context of anxiety and depression in the Ontario birth study, and the impact of maternal anxiety and depression on neurodevelopmental outcomes. Okay, so I'm going to finish with about 2 slides. And this is essentially the blood-brain barrier. And I'm going to talk very briefly about 2 transporters, P-glycoprotein, breast cancer resistance protein. Enrico just talked to you beautifully about the expression in the placenta. They're also expressed at the blood-brain barrier. And here they're very important in protecting the fetal brain. We're particularly interested, as I said, in PGP and BCRP. And this shows, I think, is one of our earliest pictures, but always, I think, one of our most beautiful pictures of PGP. This shows PGP in a brain microvessel. And what you're seeing here is endothelial cells. So essentially, you're seeing the endothelial cell that's surrounding the microvessel. And you can see here the very high levels of P-glycoprotein that are present in that. This is a fetal microvessel that's in situ in the brain. So quite high levels, and these are very protective, protecting the fetal brain from different factors. So they prevent transfer of as we heard, anti-cancer drugs, antibiotics, and even synthetic glucocorticoids. They also prevent transfer of herbicides, pesticides, and endogenous factors such as cytokines and folate. So they're very important in protecting the brain. We've done a lot of work looking at how they work, how they're regulated. We did a lot of work initially in animal models. Now we're doing quite a lot of work in the human, in endothelial cells derived from the human fetal brain. But I just want to give you a little bit, the one final data slide, and this was data that was generated by Liz Eng, who was in my lab as a PhD student. And Liz became very interested in the possibility that early exposure to glucocorticoid might actually have long-term effects on PGP function and brain protection. And so she undertook a study where she treated maternal guinea pigs with synthetic glucocorticoid in multiple courses. She took the juvenile offspring, she extracted microvessels from the brain. She then digested them, was able to take brain endothelial cells and measure PGP and BCRP function in a high throughput screening in a dish. And what she saw was really, I think, very interesting. These are female offspring whose mothers received control injections or beta-methazone. These are male offspring, control, and beta-methazone. And this shows the total PGP activity in cells that we were able to extract from the microvessels. And what you can see here is that there was no effect in females, but in the males, when we looked at total PGP, and this is actually PGP by Western blot, we saw a reduction in PGP in the microvessels. And when we looked at activity, we can measure activity through fluorescence assays in 96 well plates. And what she saw was that this reduction in activity in protein was mirrored by a reduction in activity. So these animals, I'm showing you PGP function from in the brains of animals that were exposed to glucocorticoids 25 days before. So that glucocorticoid exposure has left a long-term signature, if you like, on PGP function. And it seems to be an effect that's really confined to males, and it appears to be associated with a reduction in protection. So glucocorticoids given in late gestation may in fact be reducing protection in the brains of newborns who may be exposed to many drugs in the neonatal ICU. So this is a relatively early study, and we're still interested in pursuing this. So what I've shown you then is that the prenatal environment has a very significant effect in the developing brain, that it can affect stress endocrinology behavior. It can also potentially impact brain protection. We need to do a lot more work in this field to really understand that. I've not talked to you about all of these things, but we know that timing of exposure is critical. We know that the effects are sex specific. We know that they're age dependent. You have to look at multiple times in an animal's life. They're transgenerational, and Vasilis has done this, a lot of work looking at transgenerational effects, and much of that data has been published. And I think we've got a lot of knowledge now of the mechanisms associated with programming, and likely apply to many systems, not just glucocorticoid exposure. Obviously, I speak a lot to obstetricians and gynecologists, and many of them say, well, what does this mean in the context of us using glucocorticoids in women who are at risk of preterm birth? And the bottom line is that I'm not suggesting that glucocorticoids are not used. They are incredibly effective at reducing infant morbidity and mortality. What I'm saying is that we need to be very thoughtful about the overuse of glucocorticoids, that we don't use more than one dose or one treatment, which is 2 doses 24 hours apart. That's the standard of current standard of care. We need to think very carefully about giving additional doses. And better still, we need to identified better ways to prevent preterm birth, then we don't need to give glucocorticoids at all. But clearly there's a very significant signature associated with them. With that, I'll come back to all of the folk who did the work. I just present it on their behalf. Thanks very much. And
Yeah, you master. Yes. That means that the more experienced people in the group could master the pointer. Very good job. So, questions?
Hi, Stephen. That was a very interesting presentation. I was wondering, because you said you found some methylation markers on your birth cohort and that you follow up the infants until 5 years, I think. Are you interested in repeating the analysis so that you can maybe investigate whether these remain there after several years?
Yeah.
And do you have any markers of neurodevelopment or behavior that you could relate to these markers, maybe?
No, it's a great question. So the answer is yes. We will have the opportunity. I mean, we want to expand out these studies. In terms of looking at neurodevelopmental outcomes, most of the children, or well over 50% of the children who've gone through the Ontario birth study, we do neurodevelopmental testing at 4 time points postnatally, and we end it with neurocognitive testing, executive function, using the NIH toolbox at 5 years of age. And we have almost 2,000 babies there. So, we can go back. We did the glucocorticoid study a couple of years ago, so we could go back to the Ontario birth study and pull out now another 15 in each group and increase the size of the group. And then we can certainly look at neurodevelopmental outcomes and map it. But the sample size is relatively small, but we'll probably get something out of that. What I'm more interested in now actually is using, because it's a glucocorticoid signature, we know that stress in utero and is going to lead to a similar signature, because glucocorticoids are glucocorticoids. So we might actually be able to have a sort of biomarker of maternal or fetal stress in utero, which people are really interested in because it will allow them to then link adversity to long-term outcomes. So within the Ontario birth study, we have probably 300 or 400 now of women who have increased depression scores, anxiety. We will have the blood spots from over 50% of those, and we will have the neurodevelopmental outcomes on many of those children. So that's something that we're really interested in. In terms of going back to the babies later on, we're not allowed in Ontario. It's very difficult to get REB approval to go and do a heel stick or a finger stick for research purposes in children. But we are able to collect, in some of the kids, we've got saliva at 2 and 4 years of age. So we can see if the same signature exists in saliva. Probably will, because it seems to be very robust. So we will look at that. But interestingly, we're also now doing a big study in India where we've designed the whole protocol around looking at long-term outcomes. And we've designed a protocol that will allow us to, because we're able to collect blood at 5 years of age, in part of a bigger study called the Healthy Study, we're going to be able to look in very great detail at adversity and long-term neurodevelopmental outcomes. So we're going to track the epigenetic differences.
Thanks. Can I have a second question? Sorry.
Okay, I'll make the answer shorter.
Sorry. No, I was just wondering, because you said timing is important, and you used a model that you have a drug exposure, so you know when you have this upregulation of the system. But what about like chronic exposure? Like if you think of violence or food insecurity and other, I'm thinking like what we have here, for instance, in some of our cohorts, like how do you think this translates?
Yeah, that's a really, really good question. And the answer is probably, it's difficult to know because, you know, the HPA axis does some strange stuff if it's chronically activated, in that it starts sort of downregulate. You get these very strange phenotypes of pituitary adrenal function following chronic stress. We need to understand a little bit more what they look like, but we can look to see if there's signatures in newborns. But I would say, I mean, I don't know the answer in terms of what the profile is in these people, but I would assume that there probably is elevated cortisol in the fetus. The placenta will do a good job of trying to protect the fetus, but I think there probably will be an increased glucocorticoid.
Very amazing talk, thank you. I was thinking a little bit about the skeletal muscle because glucocorticoids are really a trophic signal. It's really classical. And also, have you looked at the motor neuron development, and if so, what happened? And the kids, are the kids more athletic or less athletic, or they, because if you have more atrophic, I think they are not going to be very good in sports. And like this, you can have, so they don't exercise, and they don't exercise, they don't have BDNF, and also impacts brain development. You can have that these circuits could be impaired as well.
Yeah. No, that, I mean, we haven't looked at that outcome. We've looked very much at neurodevelopmental outcomes. The one thing that we did show in the MAX trial, which was the biggest glucocorticoid trial comparing multiple course to single course, looking at outcomes up to 5 years of age. And one of the things that we saw in that trial was that in babies that were born at term, that had received multiple courses of glucocorticoid, that when you looked at 5 years of age, there was a significant increase, or there was increased risk of neurosensory deficits, auditory, visual. This is in the children. And so we went back to the literature and we started thinking about what that might mean, because we knew that there was evidence that glucocorticoids impact myelination. In fact, myelin basic protein is the best indicator of glucocorticoid exposure. It's super affected by glucocorticoids, and it gets decreased by glucocorticoids. So glucocorticoids are known to lead to demyelination. What we did is actually we did an experiment that was exactly built on our the MAX trial in sheep, and we showed that there was also auditory nerve demyelination in fetal sheep that had been exposed to exactly the same glucocorticoid exposure. And then in the literature, there's also studies in the sheep also showing that reduced optic nerve myelination following prenatal glucocorticoid exposure. So we think that there is some impact on myelination, which might be leading to these neurosensory deficits. The numbers weren't huge, but certainly there was an increased risk. So that we think there may be a mechanism there. Again, the outcome of that trial was give a single course, you don't need to give a multiple course.
Steve, fascinating talk. Thank you so much for your kind of your presence here, and I know you change your schedule, your trip, just to accommodate with the workshop. Really, that's the first thing. And so, just one question. I'm trying to link several aspects that we, the talks that we covered here. What is the effect of glucocorticoids on microbiota? Because so this the recent paper that showed that pregnant mice treated with antibiotics kind of destroy the microbiota. There are some specific fatty acids, short chain fatty acids. One is butyrate, which is histone deacetylate inhibitor. So that actually would change with epigenetic things. So you mentioned methylation, but I was just trying to make a connection with this.
It's a great question, and I actually don't have the answer at all, apart from, you know, the only connector that I know about is the impact that glucocorticoids have on immune function, and of course they're immunosuppressive. So what that means in the context of the gut microbiota, I don't know, but I'm sure there is an impact. You know, glucocorticoids will absolutely talk to the gut microbiota.
And the reason I thought about asking you, because you mentioned about intervention. So dietary intervention could be one way, because we have natural histone deacetylate inhibitors in broccoli or whatever, not only methyl donors, as you said, folate, but so that's kind of a, yeah.
Well, maybe that's a way of protecting it. You know, glucocorticoids are so effective. We're never going to, there's no drug company on this planet is going to try and create a better glucocorticoid, a glucocorticoid that does better targeting, because they're actually contraindicated in pregnancy. So it's never going to happen. So, you know, I think absolutely we need to think and know more and learn more.
Thanks, Steve, for this wonderful presentation. I would like to know if you have some thoughts to share with us about the mechanisms related to sex differences in the phenotype of the animal models or the human.
Yeah, we did quite a lot of thinking about that early on and then gave up. I mean, it was a cop-out, really. So we thought we'd be really clever when we first started seeing these sex differences. And I should say that when we tried to first publish this paper, our first paper in 2000, the reviewers didn't believe the fact that there were sex differences.
I believe.
So because everyone did studies in males, right?
Yeah, everything was.
So what are the mechanisms? So we got very excited. Maybe it was simple, that there was a difference in the expression, developmental differences in the expression of glucocorticoid receptors in the brain. But so we did a whole study on that, and they were the same. I mean, we didn't go into great detail, sort of looking at all of the signal transduction pathways, and maybe we should have done that. And maybe there are differences, but if you just look at receptor numbers, there aren't. But these sex differences are present very early on, because if we look at NMDA receptors in the brains of fetuses that have been exposed to glucocorticoids, and then neonates that had been exposed prenatally, we see a massive effect in females on NMDA receptor NR1, no effect in males. So they're there at the time of exposure. So it's not like they emerge over time. The big sex differences are there at the time of exposure, day 50. We haven't gone back any earlier. So that's a very interesting question.
Can I comment?
Of course.
Could be, they are all nuclear receptors. Could the surge of glucocorticoids change the receptor in a way that would interact with estrogen receptors in a way, or compete with another ligand?
Yeah, I mean, that's right. I mean, glucocorticoid signaling is these steroid receptors are quite complex, right? They all have chaperone proteins. Maybe there's sex differences in the way that glucocorticoids change the expression of the chaperones.
Right,
I mean, we look at things like, FKBP5, we look at heat shock proteins, but actually when you look at the complex, there's loads more that could be different between the two sexes. We don't know. People need to start doing work on this, and funding agencies need to continue to support this research, because it's been so underfunded. And now NIH, Canadian Institutes of Health Research, unless you consider both males and females in your research, you don't get funded. And it's
this is good.
It's fantastic.
Yeah, it's good, yeah.
But historically, everybody's done their studies in males.
Any more questions? So thank you, Steve, very much. And now we close. More slides. So I want to invite Tatiana to come here. Then we can, end our workshop. The workshop, as Tatiana told you, started in 2017. It was the first one, and Steve and Enrico and Tatiana were not here in our building, but we are here. And then we had the second one with amazing group of guests. And Tatiana was not with us because she was in a postdoc in Cambridge. And now we are here for the third one. And I think that all the 3 editions were great. Some of you were here in all the editions, so you can, just tell people how great they all are. And thank you very much to be here. And now Tatiana is going to tell you about the 4th one. It's good that we already have a plan.
I want to give a small gift for the speakers. This is coffee and a mug. So Tanya,
thank you.
And the coffee is from the
coffee shop.
Yeah, the coffee shop and the group that actually did. Yes, thank you so much. So Café del Rio. So we are very happy and thankful for actually if we could we had gifts for everyone, but so you have the notepad and also the pen.
Yeah.
Yes, thank you, Tanya. And so I'd like to first to reinforce that there are it's clear many synergies and convergent points of all the environmental factors that we addressed here, that were addressed here today, and many more that endless, of course, environmental factors that we even don't know that they will and can influence. For instance, we can say the changes in microbiota during COVID infection, right? And also maybe vitamin D and COVID and pregnancy. And also, as we point out, the effects of glucocorticoids in microbiota and also brain development, and the ongoing or the future collaboration with Enrico and the cannabinoids. And yes, so there are many, lots of things to do, and also, but things for us to reflect, right? And that's what and we'll never, it's kind of I think is almost endless subject. And we want to continue this conversation on the next workshop, hopefully next year. I don't know if I can. Yes. Okay, thank you. Obrigada, Vinícius. At the end of next year, Enrico, we were discussing that maybe biannually, but I need to have the next one at the end of next year because of the funding that we received, and hope you were all invited, and we wanted to have it in the university, but probably not on this campus, but in the campus on Praia Vermelha, or in different building. This is a Colégio Brasileiro de Altos Estudos, which is a nice building, right, and different venue. And it's in the south part of Rio. So we have the Guanabara Bay and the Sugarloaf. And so, and we want to kind of bring more researchers that are interested in other aspects of the environmental. So such as pollution, such as microplastic. We know that placenta is kind of, we found microplastic, it was found microplastic in the placenta, and also in other tissues. So that's what I wanted to say, that we will continue to investigate and to explore these environmental aspects of, the effect of long life health. And that's it. And I hope you all can come. And now we are going to end. And outside, I think there's going to be refreshment and beer and maybe, let's see. Yeah, no, outside. Outside in the café. Fora, aqui fora tem o café bem pertinho. No café. And the café, outside. Yes.
Let's end each other's lonely nights, be each other's paradise. Need a picture for my frame, someone to share my ring. Tell me what you wanna drink, I tell you what